My name is Michelle. I'm going to present on a facet of my PhD research at Monash University, Australia, on tethering RAFT synthesized SMA and SMALPs on gold surfaces. Thank you for inviting me to this meeting. In years past, I have actually joined this conference at, I think, 2 or 3 a.m. in the morning. So I'm happy to be joining at an earlier point in the evening at 5 p.m. Let me get into it now. Okay, so initially, I'd like to define the purpose behind this research. So why would we want to tether nanodisc assembling polymers to an analytical surface? So there are many benefits to polymer lipid nanodiscs, as those in this meeting will already be well aware of. So the native solubilization of membrane proteins without the need for detergent in the solubilization process, and the co-extraction of cellular lipids are some key benefits to using polymer lipid nanodiscs. And then that, coupled with the benefits to being able to tether the nanodisc to an analytical surface via the polymer belt, would involve just allow for, I guess, surface-sensitive biophysical techniques such as AFM, surface plasmon resonance, quartz crystal microbalance with dissipation, and as well as others. And by using the polymer belt as an anchoring tool, it reduces the need to directly modify or engineer the protein with binding tags, and the binding is less disruptive to the protein being analysed. So in order to achieve this goal, me and my co-authors, I guess, we used the features of RAFT polymerization, specifically the ability to functionalize the end groups of RAFT polymers, because each RAFT polymer, as some of you may know, contains an end group, and this is a sulfur-rich thiocarbonyl thio group. So here I've just got the initial equilibrium in the RAFT mechanism. RAFT is a method of polymerization, which one of my supervisors actually co-invented back in the 90s. So there's an initial equilibrium between a propagating polymer radical and a RAFT agent, which will add to the double bond between sulfur and carbon. This will produce a radical intermediate, and then the R group on the RAFT agent will cleave off, which is then capable of reinitiating a new polymerization. And then the main equilibrium in step 2 is between the propagating polymer and the dormant version of the polymer that's trapped in by the RAFT agent. So the final polymer that is yielded from this, or isolated, will contain 2 end groups. So it will contain an R group and a Z group. And these can be used, in our case, we're looking to use those as a tool for surface tethering. And there are other benefits to RAFT polymerization, as well as other polymerization methods, including the ability to produce polymers of different sequences and architectures as well. So we utilized a diblock version of SMA in this case. So the specific goals of this work were to create a library of diblock SMA polymers that had a structure that was an initial polystyrene hydrophobic block, followed by a more hydrophilic alternating block of maleic acid and styrene. This was then terminated by, in purple, you can see the trithiocarbonate group, and then a Z group that is derived from the RAFT agent in the polymerization. The reason this sequence was selected was because the more hydrophilic alternating block is positioned next to the trithiocarbonate, so that in aqueous solution, the trithiocarbonate is more likely to be exposed for binding, as opposed to being trapped within the hydrophobic core of polymer micelles. And we also wanted to target a 2-to-1 styrene to maleic acid ratio. And we compared different Z groups of different hydrophobicities. So we looked at a 12-carbon alkyl Z group, a more hydrophilic Z group, as well as a short hydrophobic butyl ZN group. And then after optimizing the smelt formation by tinkering with the sequence, we then sought to look at the effect of these different ZN groups on the ability of the nanodiscs to tether to gold, and whether the trithiocarbonate could be used for this purpose, because in previous studies, trithiocarbonates do show a strong affinity for gold, like thiols do as well. So we looked into this, and then, so here is our library of diblock SMA polymers. There's a lot of information on this slide, but the main thing to take away is we varied a few parameters. So we varied the polystyrene repeated units, so ranging from about 30 to around 12. We also varied the molecular weight, and we varied the RAFT-ZN group. And we used, we tested the efficiency of smelt formation by using a combination of phosphorus NMR and turbidity measurements using a turbidity meter. And this was after incubating the polymer at a 0.2 ratio of polymer to DMPC lipids in the form of liposomes as a model membrane system. And we found that the most efficient solubilizers were polymers that contained a low degree of polystyrene units in the initial hydrophobic block, as well as a low overall molecular weight and a more hydrophilic Z end group, with the hydrophilic version showing the greatest ability to solubilize the DMPC lipids, and it also showed the smallest and least abundant polymer micelles in the preparations. So we also removed the trithiocarbonate and the Z end group. So we used polymer D10. So this was the most optimal polymer, and we subjected this to a radical-induced reduction reaction to remove the end group. And this allowed us to understand in later binding studies the specificity of the gold binding. So this was the chemical process to remove that trithiocarbonate and Z group. So we then were able to obtain D12 using D10 as a precursor, and UV-vis spectroscopy confirmed by the disappearance of the peak at around 315 nanometers that this reaction was successful. And finally, after selecting the most optimal sequences, we then compared the gold binding abilities using a technique called quartz crystal microbalance with dissipation monitoring. And this allows you to understand by using a quartz crystal that oscillates with an applied electric current that is coated with a thin layer of gold and then mounted in a flow chamber. And to that, different solutions can be added, and the binding to the surface of the gold is monitored by the change in frequency. So according to the most basic analytical Surabhi model, the change in frequency is proportional to the mass that's adhering to the surface. So according to the Surabhi equation, there's a proportional relationship between the negative change in frequency and the mass that's being added. But this is most appropriate when the film on the surface is a rigid film. So the energy dissipation, delta D, for the future slides, is around approximately 0. And then in cases where the film is not rigid and it's more viscoelastic, it's more appropriate to use a viscoelastic model to estimate the total mass that's bound. And this just takes into account other factors like the density, viscosity, shear modulus, and the density viscosity of the bulk containing liquid, which in our case was an aqueous buffer. And these two models are important to understand the properties of, I guess, the material that's being added to the surface, as well as estimating the mass that's covering the surface. So we use this as a way of comparing our polymers and our SMULPs that were formed using those different polymers. So the best way to understand a QCM experiment is to look at the experiment itself. So I've got an example. This example is using the hydrophilic terminated diblock SMA. So this is the most optimal, most efficient diblock SMA that we looked at. So in the experiment, after getting a stable baseline, so the sensor is oscillating and the frequency and the dissipation are being measured on the y-axis. You can see after the first step, the polymer is added. So it's added at a rate of 50 microliters per minute for about 30 minutes. And then the pump was stopped. And you can see a large drop in frequency and a large increase in dissipation that indicates that a viscoelastic film is being formed on the surface. And then the pump was stopped. The pump was started again at stage 3, and the high salt PBS buffer was flown through to wash off any loosely bound material, so that at stage 4, when the pump was stopped for a second time, only the firmly bound material was captured in the measurement. So the frequency at this point and the dissipation were used to calculate the thickness and the mass coverage over time using the Surabhi and the viscoelastic models that I mentioned before. So you can see there's a difference between the two. The viscoelastic model estimates a larger mass coverage on the surface compared to the Surabhi model. And this large shift in dissipation means that, in this case, a viscoelastic film of polymer has bound to the gold. So this experiment was repeated in triplicate for each sample, and the final analysed results were compared. So we could see the difference in the mass coverage of RAFT agents compared to diblock SMA in this case. And here's just the summary of the different mass coverage estimated using S for Surabhi model and V for viscoelastic model. And then you can see that the largest mass deposition resulted from D11, which is the SMA that's terminated by a short hydrophobic ZN group, compared to SMA10, which is the most optimal hydrophilic terminated SMA, and D12, which is the N group removed. We also compared this to a commercial benchmark of lipidisc, which contains a statistical sequence of monomers. And the key findings from this were that rigid films were formed from RAFT agents and the commercial SMA lipodisc, as seen from the similar estimated mass coverage between models. And viscoelastic films were formed for diblock SMA. Mass deposition was related to hydrophobicity. So the more hydrophobic the material, the greater the mass coverage and the greater mass binding on the gold surface. The trithiocarbonate ZN group did not play a large role for polymer-only samples because the difference between D10 and D12 with the N group removed was not very significant. It was very negligible. So there's not a huge impact of that trithiocarbonate for the polymer-only samples. Whereas for the nanodiscs, the SMULPs that were assembled using the hydrophilic analog showed an increase in, or an enhancement in gold binding compared to the end group removed D12. So if you look at the empty bar on the chart on the left, you can see that in green, there's a higher mass deposition for the D10 sample. And then if you look at the graph on the right, you can see the individual QCM experiments. And in red, you can see that there's actually a 2-stage binding process for that more hydrophilic terminated SMULPs preparation. So there's an initial rapid binding that's consistent with the polymer-only control of polymer D10 on its own. And then you've got the SMULP sample, which is a mixture of polymer micelles and SMULPs, containing a 2-phase curve, so a more rapid binding and then a more gradual binding, which is thought to be resulting from the trithiocarbonate and ZN group, allowing that to be accessible for binding. And this was not repeated in the N group removed sample. So these results were sort of reinforced using X-ray photoelectron spectroscopy. So separate experiments were performed looking at the carbon content bound to a gold-coated silicon wafer after rigorously washing the wafer to simulate the conditions of the QCM experiments. And this showed the same binding trend. So there is an enhancement for the trithiocarbonate hydrophilic N-group terminated SMALPs. And then finally, AFM gave some insight into the surface morphology. So looking at the comparison between the D10 hydrophilic terminated polymer-only sample that was added to a gold surface, the approximate size range is around 10 nanometers. And whereas for SMALP samples that were applied to the surface and then rigorously washed and then measured with AFM, the particle range is a lot larger. And this is consistent with the DLS and the TEM measurements for polymer-only micelles and D10 SMALPs. In this case, they had a slightly different size. So this showed that they are depositing on the gold surface and as a viscoelastic micellular film. So the final proposed model for binding, sorry if I've kind of talked a little bit long here, but yeah, so the key conclusions were that there's a combination of non-specific binding on the surface, that is the predominant mode of binding for SMALP samples, but there was an enhanced affinity for the trithiocarbonate and hydrophilic N group terminated SMALPs. And this is thought to be due to specific sulfur gold binding. And these findings allow for a new approach in utilizing RAFT polymerization for nanodisc tethering. So thank you for listening. Sorry if I haven't left any time for questions, but I appreciate your time.
That's okay. Thank you very much for that really interesting talk. So we do have a couple of minutes for questions. So I'm actually going to jump in with one of my own first, if that's okay. I just wondered if you had any insight into why you think D10 was your most effective solubilizer out of your polymers that you tried?
Yes, so I think it's because out of all the polymers, D10, it had some of the shorter polystyrene blocks. It was in that lower range of around 12 to 14. So it had around that many repeated units in the initial polystyrene block. And I think that definitely helps because a larger polystyrene block tends to aggregate in solution and it tends to just, clump together, form larger micelles, and it's not driven to form nanodiscs. Also, the hydrophilic N group was important because comparing different polymers with otherwise the same sequence, but a different end group, that made a difference as well. So the more hydrophilic overall property helped.
Awesome, that's really good. So there's a couple of questions from Michael in the chat. Michael, did you want to ask yours?
Let's go with George's question.
Mm-hmm.
George, feel free to unmute if you wish. Sorry, I'll let you.
Yeah, sure. Thank you, Michelle, for the nice talk. I was wondering, so the surfaces that you test in your experiments are flat surfaces. Do you think it would make a difference if you switch to a spherical surface like nanoparticle surface?
Wow, yeah, that's an interesting question. I hadn't considered that actually, but I think it might make a difference, but yeah, it's hard to say without doing the experiments to confirm. But I would guess that it might create a larger surface area for nanodiscs to add onto the surface, or create more space and more binding sites, perhaps. But Yeah, that's something I hadn't considered before. So that's definitely a good question. I can't say I've got the good answer to that at the moment, but I appreciate the question.
Yeah, thank you.
Okay, so I think that's all we have time for at the
Perfect.
Thank you very much.
So Michelle, there's a couple of questions in the chat if you wanted to answer them over in the chat.
Okay, can do. Thank you.
Screen froze. Sorry, I think my screen just froze, so that was really helpful. Rosa, are you around to start sharing your screen?
Hello, everybody. Yes. Can you hear me?
Yes, we can hear you.
Right.
So our next speaker is Rosa Catania from the University of Leeds. Yeah, we're just starting to hopefully get your slides up. So she's going to be talking to us about solid-supported polymer lipid hybrid membranes. Yeah, Rosa, I will hand the floor over to you.
Thank you. Good morning, everybody, and thank you for this seminar, for inviting me to talk. And I'm going to present some of the recent work that I've been doing in Leeds University School of Chemistry with Paul Bill's group. So we work with hybrid vesicle, and the one application I will show you today is for bioelectrocatalysis, so solid-supported membrane. First, I'll give a brief overview what hybrid vesicles are. They're something in between liposome, we all know, vesicle made of pure lipids, and polymerisome, vesicle made of pure polymer. And if we look closely at the membrane of this type of vesicle, we have a mixture of amphiphilic block of polymer and phospholipids. Can you see my pointer? Just to check. Yeah, okay. And phospholipids. This one here is the amphiphilic block of polymer we have been used for most of the work I will show you today. Is a polybutadiene pack of this specific repeating units. Something this vesicle are really good at is for membrane protein reconstitution. And we have fully characterized the homogeneity of the membrane, they form a well-mixed homogeneous membrane polymer lipids. And also we look closely at the thickness of this membrane. We have recently developed a method to reconstitute membrane protein without any detergent. So we have used SMA, we isolate our membrane protein, and thanks to the elasticity of this vesicle, because of the polymer in the membrane, we can quickly reconstitute in just 30 minutes the membrane protein once we expose to a positive ion, which will crush out the SMA. And we show that the enzyme, I will not go into the detail of this work, but just to say, we show that with this method, we retain activity of the membrane enzyme once that is reconstituted into the hybrid vesicle, while we did not have any reconstitution into the liposome. So it's a very specific method that works nicely in this vesicle and can be applied even on a mixture of membrane protein of different size. And we have showed also that these hybrid vesicles have both the advantage of the two main systems, so they gain the biocompatibility from the liposome side, but they also gain the stability from the more polymerisome side. So in previous work, we show that, for instance, in a 50-50% ratio, molar ratio, we have nearly 1 year stability of the enzyme once they is reconstituted into the vesicle, while it only lasts a few weeks in liposome. So it's a powerful system for membrane protein, reconstituted membrane protein to use. And of course, we know there is a variety of membrane protein we can use, which come with a variety of application in biotechnology. Something that we are looking into is redox membrane enzyme. And those are the ones that we find, for instance, in the membrane of bacteria in this case. This is just an example. And this enzyme can catalyze lots of powerful and interesting reaction, for instance, CO2 conversion, nitrogen fixation. They are involved in lots of cycles. So we can make use of them, for instance, in biofuel cells. We can think of modify what it's fuel cells. We can modify the electrode surface, so now to have membrane with incorporated membrane enzyme, membrane redox enzyme, and we can potentially use this system to generate energy. And this, of course, sits in the need that we have for more reliable, green, sustainable way to produce energy, and stable, of course, also over time. So I will give you a brief overview of some recent work that we have done in this contest. If we want to modify an electrode in order to build a biofuel cells, we will need to consider the type of electrode we use. So we've used a gold surface. We will need an interface, which we call a self-assembled monolayer. And then we will need a membrane on this surface, which incorporate our membrane enzyme. We've used this cytochrome B03 as a redox enzyme, as a model. What this enzyme does is reduce oxygen to water while it oxidizes a quinolone molecule to quinone. And we will come back to this later in the slide, but just so you have an overview of what we want to achieve on this electrode. When we coat an electrode with self-assembled monolayer, we can use different approach. One that's been widely used also in our group for lipids membrane, pure lipid membrane, is called a tethered approach. So we have a mixture of these more hydrophilic and these more long hydrophobic molecule which contain cholesterol here, for example. And the idea is that when we fuse the lipids membrane on this membrane, we will have a sort of anchors where the membrane sits. And this approach works beautifully for liposome. What I'm showing here is some impedance spectroscopy. For those that might not be familiar, just a few important things to keep in mind. This is a called plot, and what we're looking is at admittance here. The layer that we form on this electrode act as a double layer capacitant, so we increase the resistance to the ions towards this gold electrode. So what we want to see from the SMA, once we add the lipid, is a drop in this admittance here, because we are increasing the resistance. And the lipids do this very nicely on the surface, which means we form a nicely packed insulating membrane on the electrode. We also look at some QCMD. Michelle has talked about this a lot before, so I don't need to say much about, but it's just so we can confirm that we form a flat layer and we do not have physical absorptions on this surface. So we thought it was quite straightforward. We could just switch from this system here to hybrid membrane, right? We could just have our polymer incorporated into the lipids, use the tethered self-assembled monolayer, and have the same nice reduction in admittance, but actually it didn't work. And so this surface that works perfectly for lipids does not apply for our hybrid membrane. You see we have very little reduction here, although we have some patches of flat membrane, we might not form an insulated cover of this vesicle. And what we thought was that because we have the PEG head sticking out from this polymer layer here, it might be the cholesterol was too hydrophobic and was not allowing for coverage of the electrode. So we did a screening of these two molecules in different ratio here to see if there was a good combination that would work nicely. And you see that in this case, for a 50-50 ratio, we are happy with the reduction in admittance, which says we are covering the electrode with a different type of SMA here. So we went for this ratio, 50-50 of these two short-term molecules at the interface. So now we have our hybrid membrane on this gold electrode. We characterize the quinolone molecule that we use. So I mentioned that the enzyme we aim to use oxidize and reduce a quinolone molecule. There is a variety that we can use. The one that we normally use for lipids is a very hydrophobic ubiquinone 10, which actually give us some very small reduction and oxidation peak on the electrode. If we replace this with a less hydrophobic quinolone molecule, we see that for the same molar ratio, these peaks increase a lot in intensity. So it means now this molecule here can diffuse more easily into this bilayer. So again, just to say, we have to change and adapt every single features of this system. We cannot just apply what has been working with the lipids. We have to consider now we have the polymer, the membrane may be a bit more hydrophobic and more difficult for the molecule to diffuse. QCMD showed a nice flat insulated membrane. So did also AFM. We look at the coverage of the self-assembled monolayer, and then we could see a nice coverage once we had the hybrid membrane, and there were no vesicle, but actually flat membrane. Interestingly, if we try this self-assembled monolayer now with the lipid membrane, we do not get the same nice insulation on the electrode. So the two systems, the two interfaces on the gold electrode are not interchangeable. We need to think of if we use pure system lipids or if we use hybrid membrane with polymer, we need tailored system with bespoke feature in terms of self-assembled monolayer and quinolone molecule in the membrane. Now that we have optimized this interface on the electrode and we have optimized the mediator that we want to use, it was time to see if we can actually get bioelectrocatalysis. So will the redox enzyme be inactive? Can we see current through this electrode here? So we've used our SMA reconstitution method to reconstitute the cytochrome B3 into the hybrid vesicle. If we expose this hybrid vesicle to calcium chloride, they will fuse and form this layer with the membrane protein in it. And you can see that we still retain a nice impedance spectroscopy here, which means the presence of the membrane enzyme into the surface does not affect the coverage that we have of the electrode. And this is important that we have an insulated membrane on the electrode. And we can see here in black, this is how an active enzyme looks like in cyclovoltometry, where we have these sigma tails here, and this means there is bioelectrocatalysis from the cytochrome B3. If we had a specific inhibitor for this enzyme, the catalytic activity stop, and we can see here, I hope you can see this peak here. This is just a reduction peak from the quinolone because there is not active enzyme anymore. So we regain this peak here. Something that it's very powerful, again, thanks to using this hybrid vesicle, is that we can have these enzymes stored in this vesicle for over 1 year. And then we can use this vesicle to cover the electrodes, and we can still have energy production. So it becomes to like as a potential application in the future, this vesicle can be a way to store potential energy, because then we can use them to cover the electrodes and make use of the enzyme. And so this is where we are with this project. We managed to build half of these biofuel cells, which is working. And hopefully we will have time to then look at the other half of the anode, but this is another story, not for this talk. Something I want to give you a short overview is what we are doing now. I mentioned you that we use a specific polymer for this hybrid vesicle, but we start questioning about, okay, what if we slightly change the polymer that we use? So we're in the process of doing a screening of the different polybutadiene PEG polymer. What we change is the length of the hydrophobic part of the polymer and the length of the PEG part of the polymer. We make vesicle with all this polymer. We look at cryo-EM to make sure we select the one that are forming vesicle. And we use combination of fluorophore Thank you. Yeah, we use a combination of fluorophore so that we can actually look at the orientation of the reconstitution of the reconstituted membrane protein, and we look at the size range we want with this vesicle to be in the sort of 100 nanometer size. We can see that the different polymer will affect the reconstitution. So for instance, the polymer with a shorted hydrophobic chain give us a much higher reconstitution efficiency than the one with a longer chain. We can see that different polymer will affect the orientation of the enzyme, because we have an E-tag enzyme, so we can tag the E-tag with a fluorophore and we know exactly how it's orientated. And we can also see that different polymer will affect the performance of these enzymes in terms of activity, redox activity in this case. And we are looking at many other parameters like stability of this enzyme. And we see that we have some polymer that may be more ideal to reconstitute the membrane protein, where some other polymer may give us less reconstitution efficiency, but longer time, longer stability over time, or better activity. So the idea is that we can then design an optimum system based on the application that we have in mind for a particular membrane protein, and pick up the right polymer lipids combination. So with the quick detergent-free methods we use, I show you an interesting application, I hope so, an interesting application for bioelectrocatalysis application of this vesicle, and this current screening that we are quite excited and looking forward to share that is going on. And with this, I want to thank, of course, all the PI and the groups where I work in Leeds, and you all for your attention. Thank you.
Thank you for a really, really interesting talk. It's a very different use of polymers to what I'm used to seeing, and it's really interesting to see what other things can be done with them. So we have some time for questions. Maximilian, if you would like to unmute yourself, would you like to ask your question?
Yeah, hi, Rosa. Thanks for the nice talk. Can you hear me, guys?
Yeah.
Okay. So I was wondering if you could actually also keep the hybrid vesicles intact. So I assume right now that they are basically just flattening out on the surface. So do you know a method where you can just basically keep them intact and still immobilize them on the gold surface?
Yes, you can, because for us it was actually an issue that was happening. So you can go for a different approach. One could be, for instance, try not to use calcium chloride that we add specifically for fusing them, although sometimes vesicle can just merge one touching the surface. And the other one would be to look at the self-assembled monolayer here at this interface, because the cholesterol one with the cholesterol sticking out, we were getting more sort of vesicle type absorption. And the same with the liposome. When we try the liposome on the other surface, we do see vesicle absorption rather than fusion. So I would look, if you're using gold surface, a gold electrode, try a self-assembled monolayer that doesn't match your lipids, so they will retain the vesicle. It's not going to be straightforward. There is a lot of try and error to do, but I believe potentially, yes, you can keep them as a vesicle.
Cool, that was a really, really nice explanation. Jean-Marie, do you want to unmute and ask your question?
Yeah, you can hear me?
Yeah.
Yes, Michael, yes, that's a very nice talk. My question was about the orientation of the protein in the barrier, because in your slides there, you have a unique representation. And of course, depending on the importance and the volume of the cytosolic and the membrane domain, the orientation can be quite diverse. And this can, of course, affect the data you have, because, for instance, the active site could not be on the right side.
Sorry, can you rephrase your question? Am I misunderstood?
But my question is just to know how is it oriented in the barrier?
When I looked at the orientation, I look at the, sorry if this was not clear, I look at the orientation into the vesicle, once there is reconstituted into the vesicle.
But here I try to know what's the orientation of the protein in the barrier. Okay? You can have two different orientations.
You mean, yeah, on the surface?
Yeah.
Oh, okay. So for this one here, we did not look into orientation on the surface here. It will affect the activity, I guess, but it's not something we were concerned for this project. We know we calculate how much protein is active based on the current that we generate. So that's how we know there is active protein. Then we know it's around 50% of the reconstituted, maybe a bit higher than that. So around 60% of the protein on the electrode is active. Is the 40% non-active, is not active because of orientation or other issue? We didn't look into that. Because of course, the active sites need to be reachable from the quinolone for the enzyme to work. So orientation here could be an issue, but we didn't look into this. Yeah, we are for now, we're looking into the vesicle state for the orientation. Did I answer your question?
Yeah, thank you.
Thank you.
Perfect. I think we have just enough time to very quickly go to Michael's question, unless that's already been answered.
I think it's largely been answered, but maybe Rosa, if you have any tricks for the release of the protein from the schmalp into the membrane of the vesicle, for example, maybe you can either mention them or put them in the chat space.
Yeah, you mean for the reconstitution?
Yeah, like do you use a cation level to release the protein from the schmalp?
Yes, we do. And also we do use different cation based on different membrane protein we work. For instance, this very same SMA that we use for two different membrane protein, one crush out with magnesium chloride for this cytochrome here. For another membrane protein we use, which is from eukaryotic membrane, magnesium chloride will not cause any crush out. We need to use calcium chloride. So it might be due with the tightness of the ring, I think, surrounding the membrane, and this. So
thank you.
Okay, great. Well, thank you very much once again, Rosa. It was a really interesting talk. So our next speaker is Michael-Phillip Smith from Stellenbosch University. Michael, if you want to share your screen, that would be great.
Perfect. Can you hear me at the moment, by the way?
Yes.
Okay.
So Michael is going to be talking to us today about RAFT-mediated synthesis of SMA 2-1. And I'll hand the floor over to you, Michael.
Thank you. I just want to check before I start, is my PowerPoint showing?
Looks great.
Okay, so everything's there. And there's nothing blocking the screen or anything, like a share link or anything. Okay, perfect. Okay. Hello, everyone. My name is Michael. I'll be presenting on my work for a part of my PhD so far, looking at the RAFT-mediated copolymerization of styrene and maleic acid, specifically with work with Dr. Lauren Ball and Professor Klumperman. So we all know membrane proteins, very important, used in drug binding, hormone signaling cascades, but they're very difficult to study. We previously utilized surfactants, but this resulted in a loss of the native lipid environment, which results in structural changes, and therefore impacting functionality, which is not ideal. And so we've now moved towards isolating our membrane protein, specifically with using polymers or copolymers, And essentially, by doing this, we form our nanodisc. And sort of that's how the field is going at this point. And so our current golden standard for copolymer isolation of membrane proteins is utilizing SMA 2000, which is a styrene maleic acid copolymer with a ratio of 2 styrenes to 1 maleic acid unit. So in other words, I don't know if you can see my mouse, but essentially you've got your styrene, your maleic and hydride, you copolymerize them, and then via using alkali conditions, you can ring open them and then it's aqueous soluble. The problem with SMA 2000 is it's made via conventional radical polymerization, which means it's made of a large molecular weight distribution. So you've got chains that are very short and very long, which is not ideal because you cannot know exactly how many maleic and hydride units you have per chain because you have such a large length distribution. And so you cannot functionalize a contained amount of or specific amounts of maleic and hydride units because of this large distribution. And compared to RAFT methods, you cannot functionalize a chain end. So it makes making modified SMA 2000 a bit of a difficult situation. Another thing that I'd like to have you guys think about during this talk is something called a triad distribution. So basically, your SMA 2000 is composed of triads of styrene. So you get your most hydrophobic triad. So styrene, styrene, styrene. You've got your middle ground, which is 2 styrenes to 1 maleic acid. So SSM or MSS. And then you've got your third scenario, which is your most hydrophilic ratio, which is 2 maleic acids to 1 styrene. So although you have a ratio of 2 to 1 of styrene to maleic acid, your composition can be different. And that's something just to keep into account. So we know from many studies that SMA 1:1 is too hydrophilic. And what's more ideal is you want this optimal hydrophobic to hydrophilic balance of 2 styrenes to 1 maleic acid. And so a lot of research currently, how it started, we've got the SMA 2000. Currently, we do RAFT-mediated SMA copolymerizations, and this is because now you can control the length to a specific length. You have a narrow distribution. This means you can now modify your SMA with a hydrophobic unit, so you can then mimic this ratio of 2 styrenes to 1 maleic acid to get this optimal amphiphilicity. The question I wanted to ask, though, essentially was, why not make SMA 2:1 directly via RAFT? Why first do styrene maleic and hydride copolymerization and then modify your maleic and hydride? But that comes with its own challenges and is actually an age-old task of making non-alternating SMA. So when you make SMA, you need to understand that styrene is an electron-rich monomer and maleic and hydride is an electron-deficient monomer. And so as a result of this electron-rich and electron-poor monomer, you have this perfect alternating structure. So if you have a 1:1 ratio of styrene to maleic and hydride, you copolymerize it, you get an alternating copolymer. If instead you were to now have a 2:1 ratio of styrene to maleic and hydride in your monomer feed when you copolymerize them, what will happen during this copolymerization by RAFT is you end up having an alternating copolymer of styrene, maleic, and hydride, and then you'll have an in situ formation of a polystyrene tail, which is not ideal because now you end up with a block copolymer. And if that's not what you're targeting, if you want a non-alternating SMA, this is not exactly what you want. And then you would ring open and you'd now have an amphiphilic first block and a hydrophobic second block. So what we were looking at was, instead of having an extremely electron deficient monomer, could we not use a less electron deficient monomer so that the alternating tendencies of the copolymerization are sort of removed or limited, if you would. And so we looked into copolymerizing styrene with maleic acid directly. So to start off our investigation, we went and did some computational studies. We wanted to see if the maleic acid vinyl bond is more electron rich or less electron deficient than maleic anhydride. And by using DFT computation, we were able to determine the pi bonding order, which is greater than maleic anhydride, and also the Weiberg index also indicates, since it is higher, that maleic acid is less electron deficient than maleic anhydride. So in other words, maleic acid is less of an acceptor monomer. So with this copolymer system, typically, well, when you are working with a new copolymer system or one that's very limitedly investigated, you essentially need to determine what optimal RAFT-Z group you need to utilize. So typically with styrene maleic anhydride copolymerizations, we use trithiocarbonates. But for this system, we had to see what's going on. So we screened 3 different RAFT-Z groups, and essentially what we found was dithiobenzoates are not ideal. The reason for this is the conversion is a lot lower. After 30 hours, the dithiocarbamates are also not ideal because the dispersities are higher than 1.6, which is not great. And then trithiocarbonates sort of gave the best of both worlds. We had a lower dispersity, about 1.2, and then our conversion was higher. So the rest of the studies we did essentially trithiocarbonate polymerizations. What we then did was we took the 2 monomers and we copolymerized them at various feeds. So 90% styrene, 10% maleic acid, and then you work your way down 50-50 and you swap it around 90-10. And by doing this, you can collect the copolymer from the copolymerization, do quantitative carbon NMR. And by doing this, you can essentially determine what is the ratio of SSS, MSS, and MSM fractions in the copolymer, which is quite important for later on. So essentially, you can determine the triad composition of the copolymer. So what we found via doing this is we could determine the reactivity ratios of the monomers. And what we found was very interesting. So essentially, the styrene maleic anhydride copolymerization generates a reactivity ratio, which indicates that the comonomer pair follows the penultimate model. What this means is essentially the copolymer is an alternating structure. And this confirms what we see everywhere. Styrene maleic anhydride copolymerizations are always alternating. What's interesting is the styrene maleic acid system has a completely different reactivity ratio. And so it's not necessarily always alternating. It instead follows the terminal model. And this is quite interesting because now you can form non-alternating SMA in a much more facile manner. Also, the styrene maleic anhydride copolymerization at a 1-to-1 ratio reached 0.37 per hour Kp apparent, where the styrene maleic acid copolymerization is significantly slower. And we hypothesize this is because it is less of an acceptor monomer. And so that donor acceptor pair, it's not as strong. And so it's a much slower copolymerization. So what we did then is we made a small library of styrene to maleic acid copolymers with different feeds. And we were able to synthesize 1-to-1 SMA, 1.5 styrene to 1 maleic acid, and then 2-to-1 styrene to maleic acid. What's interesting, via carbon NMR, we were able to discern that the fraction of maleic acid in the styrene maleic acid system matches that of SMA 2000. The molecular weights are very similar, but the dispersities are different. And so essentially, we have now made narrow molecular weight SMA 2-to-1. That is not a block copolymer. The maleic acid is completely, it's equally incorporated within the backbone. And then we proved this via carbon NMR as well. I'm just not showing it here. And so we did some testing to determine how similar our SMA 221 is to the hydrolyzed SMA 2000. So we did thermogravimetric analysis, and you can see here that the curves are very different, particularly down here at about 190, 200. There is this extra curve or shape over here for our SMA 221 compared to SMA 2000. And so we wanted to know what was causing this, and the answer actually came from a 1993 patent by Professor Klimperman, where he noticed that SMA, or styrene maleic anhydride, undergoes a reaction from 200 to 270 degrees Celsius, specifically forming a spirodilactone functionality as a result of MSM fraction. So basically, this reaction results in a loss of mass only at MSM fractions. And if we look over here, our SMA 221 has a higher degree of MSM fractions, which further correlates or identifies the fraction within our material is indeed this composition, and explains that we do have a higher MSM fraction. We then wanted to do some testing to see how our SMA 221 behaves to SMA 2000. If we look over here at our pH stability, you will notice that our SMA 221 is not as tolerant to pH changes. And this has got to do with the fact that we have a higher SSS or hydrophobic domain compared to SMA 2000. And so ours, if you were to lower the pH, you would create a more hydrophobic polymer, which would cause it to agglomerate quicker. And so this is why the observation is observed. Then if we look at our magnesium tolerance, everything follows the same order as you have more maleic acid units. So the tolerance for magnesium increases. But then when you get to calcium, things get a bit rocky. And so what we wanted to do was take a bit of theory from double hydrophilic block copolymers, which is looking at Z ratio. So that's looking at how sensitive a carboxylic acid unit is to magnesium or calcium per carboxylic acid unit itself. And so via doing this, what we were able to discern is that through our series, the magnesium tolerance is relatively stable. It doesn't change too much, but the calcium tolerance changes quite a bit, where as you increase your styrene to maleic acid content, so your Z ratio or your tolerance for magnesium or for calcium drops. What's interesting, though, is the difference between our SMA 221 and SMA 2000, where the SMA 2000 is a lot more tolerant to calcium compared to our SMA 221. And we hypothesize that this is a result of an increased SSS fraction in our material, essentially. And so because of this more hydrophobic domain, as we add more calcium, the SMA 221, our version, will precipitate out faster than the SMA 2000, which has a much larger component of SSM, which would stabilize it in solution. And that's why ours is less tolerant to calcium. So it's just a quick guide to thermodynamics for those who haven't done it yet. Essentially, over here we have our synthetic lipid vesicle, and then as you introduce your polymer, you have a drop in signal or turbidimetry, and then essentially you form your nanodisc, which you can confirm the size via DLS. So for our SMA 221, and all of our derivatives, actually, it solubilizes DMPC vesicles quite well. We then moved over to POPC vesicles, where essentially we found that our SMA 1:1 and SMA 1.5:1 does not really solubilize it. The vesicle size is massive, where our SMA 2:1 does solubilize. But what's interesting is that the rate of solubilization is significantly slower than commercial SMA 2000. So ours does solubilize. It just takes about 20 minutes or so. So why does SMA 2000 and our SMA 2:1 behave differently? They have the same styrene to maleic acid ratio. They both have a ratio of 2:1. They both have a similar molecular weight. So the answer to the question is essentially that the copolymers have different microstructures. That's the only thing that's really differing between them. So as a result of the different SSS, SSM, or MSM fractions, the different ratios, that has implications on how effectively the copolymer solubilizes synthetic vesicles. And so essentially, our recommendations is that copolymer microstructure is an important thing to consider when deciding copolymers, and that copolymers from monomers should rather be the option rather than modifying, because modifying is probability driven, while having a copolymer from its monomer synthesis, you're now looking at reactivity ratios, which is a bit more reliable. And so we'd recommend rather using unique comonomer pairs going forward. Thank you.
Okay, thank you so much for a really, really interesting talk. There's lots of things in there that I hadn't seen before, so that's really cool. So we do have a couple of minutes for questions. I see the other Michael has a couple of questions in the chat. Michael, would you like to ask Michael your questions?
Sure, and if anyone else has questions, feel free to ask. You're the priority here, but if there's other questions, I'm wondering about the ability to add other substituents along the backbone. Is it easier to add like a methyl or a halogen group on the styrene with your method? Would it still react equally well or better so that you can maybe sterically hinder the backbone and have a different polymer basically with possibly some advantages? Would that be easier with your method than with the standard SMA production method?
I would say having any donor groups would be ideal. Anything that's going to be electron withdrawing, I would not recommend. So any halogens, because the polymerization is already quite slow because you've now removed that donor acceptor electron distribution. If you add electrons to your styrene via donor groups, then that would probably even help it polymerize faster. And so I definitely think it is compatible. I just think adding withdrawing groups may make it practically not feasible. You'd have to like polymerize it for like 2 days.
Yeah. Do you think you'll try putting the methyl groups there? Because that would be really exciting if you could tighten up the backbone and then maybe get more resolution as a result on the proteins in the schmalps because they're more rigid, basically.
Yeah, no, definitely. I mean, there's still lots of time in my PhD, so I definitely will take into consideration.
Also, just wondering about the NMR spectra. Another big advantage is if you can get more narrow lines for NMR, like carbon-13 or proton, you can hopefully better resolve what the styrene is interacting with in particular, or maybe the maleic acid group. How much sharper are your lines, and would that be useful for looking at interactions maybe of those of the polymer with other molecules?
I haven't actually looked at myself. So the way when we did like solubilizing and things like that, we didn't actually look at NMR as a method. We just used turbidimetry and DLS. So
Okay.
that's definitely something to look into. Yeah.
But the polymer itself, the NMR lines of just the polymer are a lot sharper.
Yeah, you can. Yeah, they are fine. You can actually analyze them nicely.
Okay, great. Thanks. Okay, thanks so much.
Pleasure.
Okay, I think that's all the questions, and that's actually all we have time for. So I just wanted to thank all of the speakers from this morning's session, and I'll hand the conference back over to Michael.
Thanks so much. And our next session is going to be chaired by Johanna from the University of Helsinki. Are you in the room, Johanna?
I am.
Great.
So,
thank you for sharing.
Excellent. So if Lee-1, if you could put your slides up, that would be great.
And you mentioned also that you're looking for new recruits, is that right? for your lab?
That's right. So I recently started my lab, and we're interested in the mechanisms of how cells signal and respond to changes in their environment, particularly in pathogenic fungi. And we take this integrative approach of biochemical assays in liposomes and cryo-EM. So if you're interested in finding out more, drop me a line to get in touch.
Great. So thank you for sharing your screen, Li-Wan, whenever you're ready.
Yeah.
Thank you for coming in. You're maybe you could say a couple
Can I-
words about yourself as well. Yeah, I guess you're based in
Okay.
China.
Okay. Can you hear me?
Yeah.
Perfectly.
Yeah. Hello everyone. I'm Li Wan, and I'm now working in the University of Westlake in China. It's a very new university, and in the next month I will move to another university to set up my own lab. So I'm very happy to be here to share our previous or not previous, just the beginning of this year published our research data in Nature. So the title of my report is muscular dystrophy and dystrophin glycoprotein complex. So what's the meaning of the dystrophy? In Greek, the dys means the faulty or the difficult, and troph means the nourish. So we can clearly understand the meaning when we combine the two words together. For the clinic features of the MD, they usually work overload and work late and slowly and prone to falling before 3 years old, and around 5 years old, their motor skills will be regressed, and around 10 years old, they will lose the independent working ability, and they usually die before 30 years old. The MD patients, the MD has a very high prevalence, which is 1 out of 3,500 newborn boys with a high fertility rate. So it's very severe disease. So here I show some photos about the postures. It's very special. For example, the standing from the supine position, the cone walking, and enlarged calves. This is the normal muscle. We can see the cells between the cells are very compact, and instead, as for the MD muscles, the cells looks like very loose. And here I will give the brief history of MD. So around 200 years ago, several cases of young boys was diagnosed with progressive muscle weakness, and later this kind of disease was named as the DMD. And after about 1,000 years, the DMD gene was cloned and identified from the X chromosome, and it was also regarded as the main cause of the DMD and BMD. DMD and BMD is two different kind of MD disease. So later in the 1990s, the dystrophin was purified from the rabbit skeletal muscles, and its social protein was also purified. But later, this kind of social protein was identified as the glycoprotein. So we call this kind of complex as the DGC. In the at the early of the 21st century, the scientists focus on solving the structures of the DGC complex, but only some component or the partial full component structures were solved. This kind of structures gave us allow us to better understand the details of the component, the function of the component of DGC. So it is widely believed that the model of DGC is looks like this. We can see all of the knowing component of DGC. The DGC just contains 3 subcomplexes, which are the sarcoglycan subcomplex, the dystroglycan subcomplex, as well as the dystrobrevin and the dystrophin subcomplex. We can see from this cartoon, the sarcospan and the sarcoglycan subcomplex just locate on the one side of the beta dystroglycan, and the other proteins, just the other intracellular proteins. But these cartoon models just generated from the 2D experiment, for example, the protein-protein interaction. So it doesn't represent the real structure. So as we know, according to these models, because the up to now the clinical mutants was found to be associated with the MD. So if the MD functions is abnormal or the mutants occurs in the proteins, it will cause many kind of MDs. For example, if the mutants occurs in the glycan subcomplex will cause the LGMD, and the mutants in the dystrophin, it will cause the DMD and BMD. The DMD is the most severe MD types, and if the mutant occurs in this part, it will cause the CMD. Besides in the skeletal muscles, the DGC also exists in the other tissues, for example, the heart or the CNS tissues. The abnormal of the DGC will also cause the other kind of disease, such as the cardiomyopathy. So we all know the structure decides the functions. So many scientists just want to try to solve the overall structures, but up to the time we publish our data, only the component or the part of the protein structures have been solved. For example, the LG 4 to 5 domain from the laminin up to this part, and the CC-rich domain in complex with the dystroglycan, this part, the peptides, the complex structure have been solved. But there's no overall structure. So the lacking the information of the overall structure of DGC just limited us to better understand the mechanism of the MD. So our work is just to try to get the DGC samples and try to solve the structure and see what it look like, the real overall structures. So we propose 2 questions. How are the DGC components are assembled? And on the basis of structures, how do we explain the effect of these clinical mutants at the molecular levels? So let's focus on the first questions. So the first step to solve the structure is to get the protein. We tried many methods to get the proteins. For example, expression in the E. coli cells or the mammalian cell system, but none of them succeeded. Then we transferred to purify the native DGC from the mouse skeletal muscles directly. So on the basis of previous published data, we designed the 2 bait proteins to try to pull down the whole DGC complex from the muscle directly. And first we tried the beta protein 1, but we failed. We cannot get the obvious peak from the gel filtration. And luckily we succeed when we use the beta 2 proteins. This beta 2 protein is the LG 4 to 5 domain from the laminin up to these 2 domains. It can bind the glycosylated proteins. So we just add the flag affinity tag to the C-terminal of laminin up to proteins and mix this protein, purify this protein and mix this protein with the memory of the skeletal material. And after affinity purification and the gel filtration, we check our samples by West blot and mass, and then for the cryo-EM analysis. So this is the gel filtration result. We can see there are 3 peaks from the result, but we don't know which peak, which fraction or which peak represents the target protein DGC. So we perform the West blot to check which fractions contains the components of the DGC. So we can see the shared fractions we can contains the most knowing component of DGC. So we collect this part for the cryo-EM samples. And so we, after calculation and we got the map, the overall map, and we can see this is totally different from the previous model. In previous model, the cyclospine and this cycloglycan subcomplex just located on one side of this wheat proteins. This wheat protein is the beta dystroglycan. But in our model, the cyclospine protein and the cycloglycan subcomplex just located on the both sides of this beta dystroglycan, and the another three cycloglycan component, the beta, delta, and gamma component just forms a very rigid structure, and has a very long structure. And another component of alpha cycloglycan and the dystroglycan, they just attach to the very long structures. Later, we name these long structures as the ECD tower. So after getting the map or the structure, we can easily explain why the beta protein 1 cannot pure pull down the whole complex from the muscle tissues, because we use the system-rich domain as the beta protein. So in our structure, this protein just interact with many other proteins. So it is hard for this beta protein to replace the native proteins. So that's why we fail to get the PGC protein using this beta protein. And I will not introduce too many details about the structure, but I want to introduce some features of the structures. We can see this ECD tower is very long and it's very rigid, because on the top of the ECD tower there's charge interaction and 6 pair of the SS bond to stabilize the structure. And in the middle of the ECD tower, there are many hydrophobic interactions to stabilize the structure. So the very long and the very long structure formed by the delta, beta, and gamma cycloglycan is very stable. And we compare the similar structures published previously, and all of other structures, they are very straight. But in the case of the ECD tower, in our case, it is a little bent. The bent. So we just think what's the significance of this special bent tower. So we guess it may transfer the just to distribute the force from the different directions, so it can protect the force from the damage. It can protect the muscle from the damage. And the spring-like structures also we guess it may also have the conformation change to facilitate the mechanical transductions. But in our case, we didn't get the different conformation, just get one conformation. And another point of this project is the resolution part, because this is the original map we got, but we can see this part is very high. It has very low resolutions, and it's hard for the model building. So after trying several calculations, such as the facility 2D and multiple rounds of 3D and 2D and AU refine, we finally got the map with the overall resolution at 3.5 angstrom. Although the overall resolution is lower than the original one, but this part is, we can see the density is becoming clear, and but it can just only allow us to build the main chain of this part. We put the dystrophin and dystrobrevin here, because these two proteins are very similar. So at first we just guess which part is the dystrobrevin and which part is the dystrophin. So we perform the many conformation. The first conformation, we just used AlphaFold 3 to predict the structure. This structure is from our cryo-EM structure, and this is the structure predicted by AlphaFold 3. So this part is identical with our real structures. So this is the one conformation And another confirmation is we just extract and purify these two proteins and to perform the binding assay using the gel filtration. We can see the dystrophin and the dystrophin they can interact very well. And we also explored the possibility of GST, which may bind to the dystrophin. And we can see there's no GST bind to the dystrophin. So we confirm this two protein we built is correct. And after getting the structures, we just want to explain the effect of these clinical mutants. We just summarize the disease-related mutants and map them in the models. We can see all of mutations just exist in three areas. The first area is in the alpha-sarcoglycan. The second area is in the middle of the ECD tau, and the third area is just located in the intracellular domains. And from the structure, we can easily interpret these mutants' effect on the structures. For example, the arginine at 81 is a positive charge, and it can interact with the surrounding negative charge. Once the arginine is mutant to 16, the charge interaction will be disrupted, so it will make the protein unstable. And in the part of the EC tau, because as I mentioned, this is from the very rigid formation by the hydrophobic interaction. Once the amino acid will mutant to another, for example, the serine at 116 amino position was mutant to the phenylalanine. The side chain of phenylalanine is very large, so it will disrupt the interaction, the hydrophobic interaction. So it also will destabilize the structures and cause the disease.
Lee, you've got about a minute left, so if you could wrap it up and share your conclusions with us, and then we'll have time for a few questions.
Okay. So we just purify this domain of alpha-sarcoglycan and to try to see whether this mutant, the mutant in this protein can affect the expression or stability. We found that all of the mutants just have a negative effect on the expression of purification. We can see the monomer parts in the wild type case is very clear band and very clear peaks, but all of the mutants, the monomer peak disappeared and the band also disappeared. And in another case, we purified another mutant protein in the dysproglycan compared to the wild type, the protein is gone. So the single mutant can cause the severe MD disease. Because the single mutants can affect the expression of the protein. So here's a summary of our research. So we just got the native DGC samples from the mouse skeletal muscles, and we solved the overall structure of DGC, and you can see the interaction of each component, and we interpret the mechanism of the clinical mutants based on the structures. Here I will show my thanks to my collaborators, Dr. Jianping Wu and Zhen Yan from Westlake University, and the students of Qi Kuixu and Dr. Gao Xinhuang, and the Dr. Xiaofei Ge from Tsinghua University, and our collaborator from University of Iowa, and the facilities of West University. Thank you.
Thank you very much. That was an absolutely fantastic talk. So you have a number of questions to answer in the chat, but I'd like to start off with one question. One, how I noticed that you were initially using a Western blot to quantify the amount of complex that you actually got. So what were you using to solubilize the complex, and what was the final yield of protein that you ended up initially?
You mean the detergent to extract the protein?
Yeah, so
So
what detergents did you use, and were there particular sort of tricks in maintaining the integrity of the entire complex?
okay, so originally we use LMNT and CHS, this detergent, to extract the protein from the muscles. But after the gel filtration, the peak is also obvious, and we use the mice to identify the samples, and it's very pure, but we cannot get any 2D particles. So we screened many detergents, and finally we confirm the GDN detergent is very suitable for this case. So the final GDN detergent. Yeah.
Fantastic. So I think in the interest of time, let's move on to the next talk. But there are a number of really interesting questions in the chat. So if you have time to write out some answers and continue the discussion there, that would be brilliant.
Okay, thank you.
So our next speaker is Surabhi. Can you get your slides up, please?
Can you guys hear me?
Yes.
Okay. Can start sharing.
Brilliant. So Surabhi is a PhD student with David Drew at the University of Stockholm, and she's going to present, well, the title's there for all to see, PIP-mediated oligomerization of the endosomal sodium proton exchanger. So take it away, please.
Yeah. Hello, everyone, and thanks for inviting me to give a talk about my PhD research. And recently I graduated, and now I've started a postdoctoral position in Uppsala University. So the title of my talk is PIP2-mediated oligomerization of endosomal sodium proton exchanger, which is NHE9. A little bit of introduction about what are sodium proton exchangers. So basically these are membrane proteins which mediate exchange of protons for sodium ions. So they are found in all kingdoms of life, and they regulate intracellular pH, sodium levels, and cell volume. So these are basically antiporters which transport the substrate in a direction opposite to the driving ion. So these are basically solute carrier transporters, and they have an alternating access mechanism where the substrate is bound to the outward open state, and then it transitions to an occluded state inside the membrane, and then the substrate is transported inside the cell in an inward open position. And sodium proton exchanges, they follow an elevator model where the substrate only binds to the transporter domain. So one of the domain is static and the other domain is mobile, and the substrate binds to this mobile domain, and then there is a transition to an occluded state, and then it moves to an inward facing state where the substrate is transported inside the cell. So these exchanges can be electrogenic or electroneutral. So they can either transport 2 protons in exchange of 1 sodium ion, or they can exchange 1 proton in exchange of 1 sodium ion. So coming to the mammals, so the sodium proton exchanges that I'm sharing is a mammalian exchanger, and in mammals there are 13 different sodium proton exchangers. They are classified into SLC 9A, B, and C. So the SLC 9A are like exchangers which are again subclassified based on their localization. So some are localized to plasma membranes, which are from NHE1 to NHE5, and they have different functions inside the cell. And then there are intracellular NHEs, which are NHE6 to NHE9. And then there are SLC9B family, which is very close to the bacterial homologs. And then SLC9C, which is having an extra voltage sensing domains, which are similar to like ion channels. So they are like exchangers plus ion channels. So these mammalian exchangers, they have a and they have the dimer interface, and then there is a transporter domain, also called as core domain, which actually has broken helices where the ion kind of binds to the this region, and then it is transitioned towards the inward open state where the ion is transported inside the cell. The interesting part about these exchangers is they have very long C-terminal tail, and these tail is supposed to bind like some interacting partners such as calmodulin or CHIP1 or some lipids such as PIP2. one interesting thing about these intracellular exchanges is that they have like an extra loop domain, which is at it's above the scaffold, like the dimer domain. And these, like in the previous structures that we have done, we didn't see any density, like we couldn't resolve this region in the previous structure. So our aim was to explore what this loop domain is doing to the transporter. So these NHEs, like which are endosomal, they are actually localized to different compartments inside the cell. So if you see like NHE6 is localized to like early endosomes, then NHE9 is to like late and endosomes, and then NHE7 to like Golgi, and NHE8 also to like trans-Golgi region. And they have like different pH range. So each of these compartments has different pH, and this pH has to be regulated. So main role of these exchanges is to maintain this pH inside these organelles. And also like what we explored in the physiology, like these endosomal compartments have different pip distribution. So you will find like different pips localized to different endosomes. So if you see like in plasma membrane, you only find majorly like PI4, 5P2, whereas in case of early endosomes, you have PI3P, and in case of late endosome, you find PI3, 5P2. So we were trying to explore like what is the role of like lipids, and then what is the role of these NHEs. There's a difference of pH, so all these factors. And then we found, when we studied the structure of NHE9 before, we could see that this loop domain was not resolved, and this had like a positively charged lysines at the interface. So it's like, why these charged residues on this loop region, what they must be doing. And so we try to resolve the structure of NHE9 again with a loop domain. So for this, we try to make this exchanger a bit static. So we try to resolve it at an acidic pH, which can make it more like static. And then if the transport is less active, maybe you could capture the structure of the loop domain. So we try to do that. In some of the density, we could see like there is some flexible loop, but it was really hard to resolve. And then we selected like some particles which had this loop, and then they did a 3D variability analysis where we clustered these particles and then tried to resolve like this 100,000 particles further to get the structure for the loop. Yeah, we did see the loop. The resolution was not that good enough, but at least for the, like when we do the focus refinement for the loop, we could see like there is like a domain swapping between these regions. And then the AlphaFold came, and then we tried to build a model using the AlphaFold, and we could see like when you make an AlphaFold like dimer and we try to fit in the densities, you could see like this is domain swap. Like yeah, so like one of the regions of the loop domain is facing like close to the other protomer, so they are like flipped in the region. And we could interestingly see that the lysines that we found, which were there in the sequences, they are clustering towards the dimer interface. So it built us like a curiosity, what are these lysines and positively charged residues doing with the dimer interface. So further, we try to resolve the structure of NHE9 with the lipids. So for this, actually, we try to put some PIP lipids because they were known to function or regulate these exchanges. But unfortunately, we couldn't get any structure with these lipids. So what we did was we enhanced our purification with crude lipids, and this crude lipids picked up the necessary lipids which were which would be required to stabilize the dimer and also to the loop domain. So when we had this structure, we could see like there are some lipid densities at the dimer interface, and we could see that the positively charged like the lysines, they cluster above these lipids. And initially we thought that the lipid would be the PI4, 5P2. So we were trying to model that, but we could see like it didn't show, like the density was more clear for PI3, 5P2. And it also physiologically made sense because NHE9 is localized to late endosomes, and the major lipid found in late endosomes is PI3, 5P2. So which all made sense. So to further confirm this, we did this assay, which is like optimized in our lab. It's called as thermostability assay. We use a GFP purified fusion. We add lipids to it, and then we incubate it for like 30 minutes, and then we add a detergent, which is beta OG. So whatever protein is like aggregated, that can be precipitated using this detergent. We heat the sample at increasing temperature, like from, let's say, 10 degrees to 80 degrees, and then we spin it for almost half an hour, and then we pull out the sample, which is non-precipitated, like the like the non-precipitated sample, and then we measured the fluorescence intensity for that. And we could see like when we measured the fluorescence intensity, the TM that we calculated for the purified fusion was around 51 degrees, but like if you add lipids to it, the TM shifted, and the new TM for with PI45P2 was around like 59 degrees, and for 35P2 was 66 degrees. So 35 was more like stabilizing compared to 45. And so that delta TM shift was around like 8 degrees for 4-5 and 15 degrees for PI35P2. Again, like what we do is like we take this sample again and we can inject it on FSEC to see the quantity of like monomer and dimer present in the sample. And we could see like PI35P2 was stabilizing the dimer compared to 4,5-P2 and the other lipids such as POPC. So we could clearly see that the lipid which is preferred by this protein is PI35P2. Again, like to measure the activity for this transporter, we use this assay called a solid state membrane electrophysiology assay. And we make proteoliposomes first. So we take like crude lipids such as East polar, and then we add like detergent, membrane protein and the liposome. We incubate for some time, and then we do a filtration, and then we get this membrane protein inserted into the liposome. So we get proteoliposome. We put it on a gold sensor, which I think was already discussed by Rosa, like how this solid state electrophysiology experiments work. So we did with the similar assay, we had the setup, everything. And then what you do is like you add a non-activating solution. So in this case, like sodium is the substrate for this protein. So you would take any ion like lithium, or like we use choline chloride, which has a similar charge as sodium, and but doesn't it's not the substrate for the protein. So we add that as a non-activating solution. Then we add the substrate, which is like sodium, and then again a non-activating solution to have like an off signal for this. And in our case, like for these exchanges, we say that it's not calculating the actual Km, but it's calculating the Kd, like binding affinity, because even the binding of the substrate can drive a conformational change in the protein, and this change can also give a change in the membrane potential, which can be measured. So this gives like a transient current, but not the actual transport. So we calculated the binding affinities in the presence of this lipid. So what we do is like we incubate the protein with this lipid, and then we incorporate this whole sample into the liposome. So the lipid is already bound to the protein, and then we measure the signals for each of the samples. So when we measure the signal for the wild type without any lipid, we could see like the signal was around like around 3, the peak currents that we got. And then when we added this PI35P2 lipid, like the signals were higher, like around 7.5. And then we also made a variant for this, which is a dead mutant, which we call. It's the mutant which is it's the residues which bind the ion, and we mutated both of that. And then we measured the signal for this, and we could see like the signals were like very low. So we could say like this setup is now working fine. And then we measured the binding affinities for this. So the Kd that we got for the wild type was around like 36 millimolar. But when we added this PI35P2 lipid, we could see that the Kds were like around 9.3 millimolar. So which was like this lipid was enhancing the activity, or let's say binding affinity for the protein, for the ions. And then we also added PI3 and 4,5P2 lipids, and we could see like with addition of 4,5, the binding affinities were around like 4.7 millimolar, which is very close to what we get for the wild type. So this lipid was not actually enhancing the activity of the protein. So to further confirm with our results, we did some MD simulations in collaboration with Lucy. So what we did was we put this lipid at the dimer interface, and we tried to measure, like, see if these lipids like make contacts with those positively charged lysines and the arginines around. And we could see like this, the lysine 105 was directly interacting. And when we mutated those lysines, we could see that there was no contact between, like, interaction happening between these residues. Then we also measured in the presence of 4,5P2, and it could clearly show that this protein preferred PI3,5P2 based on the contact probability. So it was very clear that this is the lipid which is binding to this protein. And again, like, so as we know that PI4,5P2 was the major lipid at plasma membrane, so we tried to express this protein at the plasma membrane and measure the activity of the transporter. And we could see like NHE9 did not have any activity at the plasma membrane because there's no PI3,5P2 lipid present in the plasma membrane. So this protein is inactive when it's localized to plasma membrane. And then we measured the activity of the protein at the endosomes, and we could clearly see that there is a pH change when you have it expressed in the endosome. So this was also done in collaboration with Daniel Fuster. And from this, like, we build a model where we say that when NHE9 is expressed in the plasma membrane, there's no PI3,5P2. And this protein is very, like, this loop domain is very flexible, and because of that, like, there's no transporter is like less active. But when we express in endosomal membranes, you have this specific lipid, and it binds to the dimer interface, stabilizes the protein, and then it also enhances the activity of the transporter. And with this, I would like to conclude my talk, and I would like to thank all my lab members and my supervisor David for making me work with these exciting projects and giving me opportunity to explore about these lipids and membrane proteins and very fun projects. Yeah.
Brilliant. Thanks very much. This project is a great example of how lipids really drive the activity of specific ion channels. So we have a number of questions in the chat. Jean-Marie, would you like to
ask? Just a question about, first of all, I want to congratulate you for this impressive works. I was surprised to read and to see that POPC seems also to stabilize the dimer formation. So I was wondering whether you can separate the contribution, a specific contribution coming from PIP2, and the organization of the lipid like DOPC.
Sorry, I just, can you repeat the question, because I...
In one of your slides, showed that POPC seems to stabilize the dimer also.
Yeah.
This is not a big effect, but there is some effect.
Yeah.
So my question is, could you agree that there is a contribution coming from a very specific interaction between POP2 and your protein and organization of the lipid?
Yes.
Which is a more global effect.
So with this specific lipids, there is like these ones, because we in the structure, and we also did some native mass spec before, and we couldn't see any of the other lipids before, like the POPC. It was only this 1 kilo Dalton adduct, which we always saw in native mass spec. So it was very specific that these lipids are binding. And with POPC, in the structure, like in the FSEC, you could see like some dimer stabilization. But I think so in this assay, what happens is like the POPC might be destabilizing, but there is some proportion of the sample which still retains that dimers and monomers. So I think that's the signal that we see in the FSEC for POPC.
Thank you.
Thank you. Umut, would you like to ask your question while you're getting your presentation up and running? I don't think we can hear you.
So you showed already this cryo-EM structure in the beginning, but I guess I missed. So did you use this PIP3-5 lipids in your structure, or because you didn't see that moiety like nicely resolved, right? Maybe, but was it in the presence of this lipid?
Yeah, so what I can say is like initially when we were trying to, because at that time only PIP2 or 4,5P2 was the only known lipid. So we were trying to get the structure with that. But I think because it was not right lipid, that's why we couldn't resolve the structure before. But later on, like we added crude lipids. And yeah, we do see like, I mean, it won't, the overall resolution of that structure was around like 3.1 Angstrom. Though the densities were not very clear, but like the head groups, we can clearly see differentiate between the 3,5 and 4,5. So yeah, that was the reason that we have the 3,5P2. But we didn't add PI 3,5P2, like we didn't substitute. It is just picked up by the protein while purifications from the crude. Yeah.
Excellent. Thanks again for your talk. So if you have questions for Surabhi, please continue putting them into the chat. And next we have Umut Günsel, who is a postdoc with Franz Hagen at the Helmholtz Center in Munich. And he will present work on the structural basis of the apoptotic induction by the mitochondrial voltage-dependent anion channel.
See my slide, I hope.
Yes.
First of all, thank you for inviting me to this nice conference. I mean, as Joanna said, that I'm working as a postdoctoral researcher in the lab of Professor Hagen, and in our lab, we work on structural biology of membrane proteins. And today I will talk about our recent paper on structural basis of apoptosis induction by mitochondrial redox. And actually, this work is already in bioRxiv if you are interested, and also it's at the moment under revision in Nature Communications. As you may already, as you may already know, redox is found in the mitochondrial outer membrane, and its main function is transport of small molecules across the outer membrane, like ATP, ADP, or metabolites, or small ions. So this protein is comprised of 19 beta strands, making this nice beta barrel channel, and also it has an N-terminal alpha helical domain, which is residing inside the pore in the canonical conformation. So this protein, in addition to its main function as a transport of molecules, it has been also shown to act as a key player in the apoptosis mechanism. So during apoptosis, upon an apoptosis signal, mitochondrial outer membrane gets permeabilized, and the cytochrome C release from the intermembrane space actually leads to cell death. However, how redox is really mechanistically regulating this process is not really clear. In the literature, it has been clearly shown that the redox gets oligomerized upon like different variety of apoptotic stimuli, and also it has been shown that this protein can interact with pro and anti-apoptotic proteins, and this N-terminal helix is actually very important in this case. And even like for the oligomerization of this protein, actually, it has been proposed that the oligomerization might be directly having a role in the formation of this pore formation in the outer membrane. However, until now, we don't have any structural proof for such a conformation. And also recently, actually, in this year, an AFM study has been shown that VDAC can get VDAC is oligomerized in a lipid state. However, unlike in this proposed state, in between these oligomers were actually filled with lipid molecules. So this actually proposed mechanism might can be questioned actually with this paper. And in our story actually begins with, so what we think that since this N-terminal domain of VDAC is binding with the pro-apoptotic or anti-apoptotic proteins, it needs to be exposed to the outside of the pore. However, such a structure until now has not been shown. And what we think is this oligomerization of VDAC in the membrane might be a trigger for such an exposure for the N-terminal domain of VDAC. And in our story, first we wanted to probe the helix exposure to outside of the molecule barrel. So we used a single cysteine mutant at the very end terminus of the protein, which is normally inside the beta barrel. And by adding of a maleimide molecule, which is 40 kilodalton in size, so it should not access when it's inside the pore. However, when it's exposed in the solution, then this molecule can modify this N-terminal region easily. And actually, fortunately, we were able to, in the end of our screen, we were able to find a condition that a negatively charged cholate as the detergent actually significantly increased this exposure in the exposure of the N-terminal helix to the outside of the pore. And as we predicted, this event was actually quite coupled to the oligomerization of the VDAC as monitored with this chemical cross-linking, amino-specific chemical cross-linking experiment under same conditions with this negatively charged cholate detergent. And then as a control, we also used another mutant on top of this T60 mutation. So there is, in VDAC, there is a glutamic acid residue at this membrane-facing side of the channel, and mutation of this residue to a valine, so more hydrophobic residue, actually increases the stability of the beta barrel channel, and then this keeps and this actually decreases the oligomerization tendency of the protein, as you can clearly see here. And as confirmed with this experiment, actually, what we also observe is when the oligomerization is reduced, we also see a reduction in the exposure of the N-terminus of VDAC also in this mutant. And of course, detergents are not the native environment for this protein. So we wanted to do the very same experiment also in liposomes. So here we used POPC as a lipid, and we observed actually very similar result in the presence of E73V mutation. We observed the modification in this of the N-terminus is slightly reduced. However, this time the effects of this exposure and also the oligomerization was not as pronounced as in the detergent case, but we see actually similar trends for this. And again, to mimic the cholate, actually to come more close to the cholate state, we also added a negatively charged lipid, POPG here, in our liposomes, and we found that in this case, actually, the exposure of the N-terminal helix is increased in this case, actually confirming our model that oligomerization of VDAC in the membrane is inducing the exposure of its N-terminus, and actually negative charges on the lipid surface seems to have a stabilization effect in this conformational change. Then we wondered actually with a structural tool if we can really monitor this exposure of the helix or not. we wanted to use our nanodisk technology as a tool to for conformational trapping of VDAC in a helix exposed state. And for this, what we did, we used like different sized MSP nanodisks and checked the helix exposure again with this PM40 modification assay. And what we found here is this larger nanodisk, which give like enough space to VDAC for the for its like canonical helix inside conformation. And the smaller nanodisks generated a more confined environment, and then the helix goes out of the barrel. And actually, we think that this smaller nanodisk is actually mimicking the oligomerization state of this protein for our and we can use this for our analysis for our experiments. And first of all, we wanted to check this with NMR with a specifically methionine, specifically methionine labeled protein. And we have also an additional methionine mutation in the very end terminus of the protein, and also these two native methionines on the wall. What we observe with the larger nanodisks, actually this protein is present in two conformations. So we see for this 155 and V3M methionines, we see two different conformations. And addition of BCL-XL, which is an anti-apoptotic protein, as a partner of, as a binding partner of the N-terminal part of VDAC, we observe only slight changes in one of these conformations. One of these conformations showing that this VDAC is found as in a conformational equilibrium in larger nanodisks. However, when we use the smaller nanodisk, so then we just see only one conformation in this case, and here the peaks are a bit broadened because of the instability. Sorry, because of the interaction between the MSP belt and also the membrane protein. But what we see basically is in this time addition of BCL-XL showed actually a direct, so the line broadening in this N-terminal mutant. And as I said, it's just one conformation. So it looks like our biochemical experiments actually confirmed by our NMR experiments. And as a next step, we wanted to see these particles also using cryo-EM. And in cryo-EM, when we use this large nanodisks, VDAC is we found VDAC either as in a monomeric state or in a dimeric state in these nanodisks. And interestingly, in both states, we find an electron density inside the barrel, which can nicely which you can nicely see in this figure. However, when we used smaller nanodisks, we could not detect any electron density for the N-terminal helix, which suggests and also confirms our biochemical and NMR experiments for the exposure of N-terminal helix. Okay, now actually we for the first time showed the helix exposure state is actually exists with a structural method, and we wanted to check if this outstate, the helix outstate can bind to its interaction partner, be anti-apoptotic BCL-XL. And here you can see the NMR spectrum of BCL-XL, and addition of this smaller nanodisks on top of this on this sample actually induce chemical shift perturbations at specific residues in this protein. However, in the larger nanodisks, we do not see this effect as much as in the smaller nanodisks. And when we map this chemical shift perturbations on the structure of BCL-XL, what we observe is actually these chemical shift perturbations are clustered around the BH3 binding side of BAC. So BAC is a pro-apoptotic protein. It makes pores on the mitochondrial outer membrane upon apoptotic signal. And actually, to check this, we did one more control experiment. So we repeated this experiment, but in the presence of a BH3 helix of Bach. And what we see here, as in the larger nanodisc case, we did not see any change supporting that, okay, this BH3 binding site of BCL-XL is competed by VDAC N-terminal domain of VDAC and also the BH3 helix of Bach. Then we would like to, we wanted to get a high resolution structure of this complex. However, the affinity of this helix to the BCL-XL was in low micromolar range. I mean, it's still a good binding affinity. However, for the crystallization experiments, we utilized the fusion construct that's made where the VDAC N-terminal domain is attached to the BCL-XL C-terminal part. And this actually yielded a very nice crystal structure at high resolution, showing this complex and also confirming our NMR experiments, NMR chemical shift experiments. Then afterwards, to see if this complex is really physiologically relevant or not, we did a short molecular dynamic simulations to see in a lipid membrane environment setup. So what we see here, so this is our structure, and the beta barrel is in the lipid membrane. And as you can see in the complex formation, the C-terminal tail of BCL-XL is close to membrane because normally that has a transmembrane segment, and this looks like feasible for under physiological conditions as well. And of course, afterwards, we wanted to also check the via functional assays. So here you see a pore-forming assay, which is used widely in this for this apoptotic proteins. And Bach, as a pro-apoptotic protein, actually makes forms at high concentrations, and it gets auto-activated. And you can see this nice pore formation as the increase in fluorescence. And addition of BCL-XL, which is an anti-apoptotic protein, which inhibits Bach under normal conditions, this pore formation is actually attenuated. And interestingly, and as we expected, addition of N-terminal peptide of VDAC, actually we observe this inhibition of inhibition in a concentration-dependent manner in this functional assay. And of course, this effect can be also through the Bach directly. So in a control experiment we checked if Bach in the lower concentration needs to be activated with activator proteins such as CBIT. And we checked if VDAC1 N-terminal domain in excess amount can activate pore formation or not. And it looks like it cannot. And this actually shows already that VDAC looks like to directly affect BCL-XL inhibition, so inhibit the BCL-XL inhibition of Bach pore formation. Actually, with that, I come to our final model. So VDAC actually, it looks like, unlike it's said in the literature from our model, VDAC does not have a direct role in the permeabilization of the outer mitochondrial membrane. It's rather act as a sensitizer, a BH3 protein to induce apoptosis. So under normal conditions, the protein is in a monomeric state. Monomeric state where the helix is inside. However, in the when upon like apoptotic stimuli, mitochondrial damage, and upregulation, so VDAC gets oligomerized, which we don't know how this is really regulated, but this requires further investigation. The N-terminal helix of VDAC can get exposed to the cytosolic side of the mitochondria, and this competes with Bach for BCL-XL binding, and in the end, Bach stays free when the BCL-XL is inhibited and the pore formation occurs. So here, actually, VDAC is, as a sensitizer protein, is inducing apoptosis upon specific signal. And with this, I would like to thank you for your attention, and also our lab members and also all the contributors, our cryo-EM collaborators, and also for the crystallization, our collaborators in Helmholtz. And yeah, if you have any questions, I'm happy to answer.
Brilliant. Thanks for that talk. We do have a few questions. George, do you want to ask your question?
Thank you for that interesting talk. So you show with your detergent experiments that the exposure of the helix depends on the environment it's in, and you speculate that in the mitochondria this is I think it was negatively charged lipids. How does that fit in with this demonstration that when VDAC1 oligomerizes, it can mediate lipid scrambling? So it could potentially, basically, you might lose any asymmetric lipid distribution that you had. So how do you get this concentration of negatively charged lipids to induce this extension of the N-terminal helix? I don't know if that makes sense.
I mean, I am not sure if I understood correctly, but so normally VDAC is in a dynamic equilibrium between the helix in and out state. So this happens, and actually what we think is the negatively charged lipids are enhancing in one of the conformation. It helps in one of the conformation when the VDAC is oligomerized, because we also see that in the okay, sorry, how do I go to? Yeah, we see that also in the monomeric state, we see to some extent the exposure of the helix. However, in the presence of this negatively charged lipids or detergent, we see an enhancement in this exposure. So could I answer your question, or?
It does help a little bit, yes. I think the next bit, yeah, you said you're not sure how this is regulated. Well, I guess that would be the next bit. But yeah, but thanks, that does clarify a bit.
Yeah, exactly. I mean, how the oligomerization is really managed, this is not really clear, but it's known that under apoptotic condition, VDAC oligomerizes, and also the negatively charged lipid density can change upon this oligomeric signals. And these are all probably the all leading causes for these effects that we see. Yeah, because also another thing is during this apoptosis formation, that VDAC is proposed to maybe release the cytochrome C from the channel. However, actually, cytochrome C is like a 12 kilodalton protein. It's very hard to pass through the channel itself. And also such a structure doesn't look really feasible, because then how the lipid molecules in between the monomers are excluded from making a bigger channel. So that's also doesn't look very energetically possible. And so actually our mechanism is more indirect, like making the backbore formation in a secondary effect.
Brilliant. So in the interest of time, I'm going to thank Umut, Surabhi, and Lee again for their talks. There are still a number of questions in the chat, so please feel free, speakers, to look through those questions and answer what you're able and take the questions to the chat. And with that, I think we have a minute or two of a break until the next session starts.
Okay, thank you so much for chairing and all of the great talks. I do want to ask you for a favor, all of you. If you could vote for the best talk, that would be great. And I'm just going to launch that in a second here. And just consider also the ones at the beginning to give your vote. Here we go. And then based on that, we'll decide the award, which is from Nanoscene, which is one of our sponsors. So thank you to the Nanoscene group. And thank you to all the speakers. We do have one more session. So again, thank you, Johanna, for chairing, and also Becky beforehand. And if Naomi is available, maybe you could chair our next session as well. That would be great. And I'm just going to maybe unshare your screen there if I can, and ask Naomi to come forward.
Hi, I've just turned my video on. Hi, Michael.
Oh, hi there. Nice to see you.
Nice to see you.
If you want to introduce yourself, and if you're looking to recruit anybody, you've also got a newer lab. So if you have any positions open, feel free to let the people in the room know. And any other news you want to share, and then let's kick off when you're done with our first speaker.
Great. I'll keep it quick. I started my lab just under a year ago at Aston. I don't have any positions open. I'm hoping to put in some grants as soon as possible to look at kind of fundamentals of SMALP formation and stability. So yeah, hopefully we'll have some news on that over the coming year or so. Anyway, I think we should. Sorry.
You've got a conference next April as well, right? Do you want to mention anything about that?
To be honest, I'm not really involved in organising that. I think that's more Alice, my colleague at Aston, Alice Rothney, I think, is involved in organising that. So I'm sure she will advertise it through SMALP.net when we can. I suppose I should say we always organise a membrane protein workshop in Birmingham every year. So if anyone is interested in either putting forward people for that or coming to it, then that would be great. It's normally popular. We're trying to set up a second session of it to get more people trained. So, okay. Let's go. So our first speaker in the session is Eamonn Reading. I'm really excited to have updates on what he's been doing because I've been sort of seeing his work over many years now. So Eamonn, are you ready to go ahead?
Yes, I think. Right.
Great. I mean, no rush.
Yeah. Is this all shared?
Yeah.
Everyone can see this? We're all good?
Just full screen it. Good. Perfect.
Yeah. Okay, cool. I didn't know there was going to be prizes, so I was asked to give this talk. Me and Ryan duly were asked, and I've kind of taken it upon me. But I've basically taken a prize away from you, Ryan. So I apologise because Ryan really has been leading this in his PhD. This is the kind of bulk of his work. So I'm just going to try and do it justice. Maybe I'll buy you some lunch or something later. So obviously, it's kind of obligation to start with some scary numbers, right? That's just what we have to do. Say we're doing something important. But we're all pretty much aware of antimicrobial resistance. It's the kind of silent pandemic. And, we haven't invented a new drug for a long time, although this is getting better, to be honest. Pharma are now getting back in the mix. Roche has, got a new drug and things are looking promising, but it's still a issue which we need to face because we no longer want to die from pricking our finger on a rose bush and getting an infection and that be the end of it. So we want to understand how bacteria, in particular gram-negative bacteria, kind of battle antibiotics, how they become resistant. And because we're membrane focused here and because our whole lab is obsessed with membranes, we kind of focus on the mechanism of efflux pumps. So obviously bacteria can become resistant to antibiotics in lots of different ways, but a major player and almost a kind of first point of defense, as with anything that the membrane is, that first point of defense, is to kind of spit antibiotics back out, right? So they try and get into the bacteria. You have these kind of nanoscale vacuum cleaners, and they kind of spit it back out again. So that effective concentration can no longer be reached. And it does this for a variety of different kind of molecules, but also for other kind of attacks, kind of other kind of biological bacterial warfare that's going on. So, understanding how they work is also useful because if we can stop them from working, there's a chance that you can kind of rescue efficacy of antibiotics, which we already have. We have some pretty good ones, although they're developing resistance. But also any new drug is going to face the same challenge, right? So being able to, consider synergy of stopping these pumps at the same time as develop antibiotics is something which could be quite useful to do. So multi-drug efflux pumps come in all kinds of different shapes and sizes. Here's a kind of collection of them. The one that we really are interested in is this RND type. And these are ones which kind of bypass an entire envelope. So they're involved in gram-negative bacteria where you have an inner membrane and an outer membrane. And they kind of are tripartite. So they have a kind of transporter connected by a kind of periplasmic linker protein, which connects it to an outer membrane porin like Tol-C. And what we're really interested in is, maybe if I can get my pointer up. Let's have a look. Is this a pointer? Annotate. Aha. Is the kind of transporter region. This is a real kind of powerhouse. This is where drugs are recognized, taking advantage of the protein motive force. It kind of powers this kind of motion to kind of export the drug out. And the reason we're interested in these is because they're very promiscuous. So this, this is a paper from 2013, but the types of molecules they spit out is vast, right? Chemically very diverse, right? They can be greasy, they can be polar, they can be long, they can be lipidic, they can just be simple, all kinds of molecules, right? So they really are the kind of waste disposal mechanism, the kind of real kind of vacuum cleaners of gram-negative bacteria. And the reason I quite like them is they do it in a quite a dramatic way. They don't just kind of pump it in like a PGP way where they like open and close and it kind of diffuses out. They kind of machine these stuff out, right? So using the beautiful studies on the ACR-AB Tol-C complex, which is a kind of prototypical complex, these R&Ds, people have started to understand that they go through these kind of asymmetric functional rotation mechanisms where they go between loose, tight, and open states. And they kind of squeeze the molecule into the back of a distal binder pocket, and then it kind of goes through a hole at the back, and then it kind of exports up through. And all the while it's doing this, all these proteins are kind of moving around. So we find them really interesting because they're so dramatic. But we also find it interesting when you look across the types of pumps and when they kind of come into action. So like I said, the ACR-AB Tol-C system is really kind of the most clinically relevant because it's kind of always there. Right, it's constitutively expressed in E. coli and in other kind of bacteria as well. There's homologs, orthologs, and stuff. But it's by far the only pump that's there, right? So there's 20 other efflux genes in E. coli, for example. And you can see when you go into different physical environments, some of them are going to kind of switched on. So you go into like low pH, you get these other pumps switched on, but ACR-AB also kind of gets switched on as well, kind of upregulated in that way. So it's kind of, not just responsible for antibiotic export, but also other kinds of physiological responses. But what's interesting is when you consider other kind of stressful environments for bacteria. So anaerobic, so E. coli is a facultative anaerobe, right? Low oxygen, or when bacteria just get a bit tired, right? When the nutrients kind of run out, they go into stationary phases or they go into biofilms. What we start to see is you get this efflux pump very uniquely, this MdtF efflux pump uniquely gets upregulated. 20-fold in low oxygen, 41-fold in the late stationary phase. And this is quite interesting when you consider, the journey, enterotic journey of E. coli, going from your mouth down to your stomach through your intestines, as acid is going up and down, oxygen levels are going up and down. So these pumps are kind of getting turned off and on. And there's a little taster here of, what else kind of happens as the membrane also changes, but I'll go to the next slide. And here's a lovely figure by Ryan who's shown what kind of transitional regulators turn these things on. But essentially, low oxygen, low pH, this kind of pump kind of comes in. So the question is, it's really similar to ACR-AB. So why have it in the first place, right? And so when you consider, for an example, going into late stationary phase or other stress environments like low oxygen and pH, is the whole membrane in gram-negative bacteria kind of gets remodeled, right? I think this is something we need to appreciate is you start to get incorporations of different types of fatty acids, little triangles, which I think is fascinating. You get these little triangle fatty acids, these kind of cyclopropane fatty acids, which both, in a similar way to cholesterol does, both make it kind of stronger, but also kind of a bit more fluid in places, but also less fluid in other places, a kind of complex physiochemical kind of behavior there. And you also get this kind of reinforcement of the outer membrane, right? So the envelope becomes just much more hardy. So really what's controlling antibiotics or other kind of exterior toxins getting in is kind of the membrane shutting up shop, really. So it's not really the efflux pumps in this case, which is providing that first barrier of defense. In the kind of log phase of growth, ACR-AB is really that kind of thing because this membrane is a bit more pathetic. Things can kind of get in and through, and that's important. But when you really get to these kind of more stressful states, it kind of shuts up shop. So the question was, why do we need this MdtF expert? Why is it made in those environments where the envelope really is providing the main barrier to attack? And so we thought it was important to capture MdtF and to understand what it's doing in a native lipid environment. So MdtF hadn't had a structure beforehand, even though it's very similar to ACR-B. And ACR-B has kind of really been the kind of gold standard for studying things with SMALPs, right? So we thought, we'll try SMALPs. We were fortunate enough where we got a bunch of free SMAR back in the day from the company before they commercialized. So it became relatively straightforward for us to do this. And we used SMAR 2000, and Ryan went forward and we got, really nice purified protein. We use a kind of his tag on the, kind of standard IMAC kind of purification by size exclusion as well. And we got really nice protein. And then we did something which I've never done before. I'm a structural proteomics lab. I usually like mass spec, but Ryan kind of took this upon himself and he did cryo-EM, did it in collaboration with UCP Pharma. And he got lots of lovely particles and we could see the discs there. And he finally got a resolved structure of MdtF. And when we look at it, we can see it's in its native lipid belt. And what's really nice, as seen with ACRB, is it's a kind of homotrimer. So you have these 3 different protomers which go through those asymmetric states. And because you have this kind of pocket in the middle, you kind of get some resolved lipids in here because it's kind of, trapped, kinetically kind of trapped from moving around too much. So we can kind of see that we get lipids. This is kind of a lipidated disc around this protein. And we see the, the usual suspects for E. coli, PE, PG, cardiolipin. But what was interesting is when we said GCMS, we could actually see, a decent enrichment of cyclopropane fatty acids. So these little triangles are in there as well. The reason you can't really see this with fragmentation, LCMS and lipids, is because they fragment the same way as a double bonded fatty acid would, because of the same kind of amount of molecules, atoms there. So really GCMS is a way that you can kind of separate these things. So it's worth bearing in mind that maybe your stationary phase expressed proteins might also contain CFA. So bear that in mind. And then we had the cryo-EM, so we had like the ground state, what MdtF looks like, right? This kind of frozen ground state of the protein. And of course, because we're kind of thinking, why is MdtF there and not ACRB, right? And ACRB is this kind of prototypical. We always kind of compare it with ACRB. And ACRB and these R&D proteins are kind of split, like I said, into these transmembrane domains, which are where the proton motive force powerhouse kind of powers how these proteins move. You've then got this porter domain, which is where these kind of molecules are recognized and the binding pockets are. And then you have this funnel domain, which is what kind of connects with the periplasmic domain proteins to connect to the outer porin. So you kind of split them into these 3 areas. And when we look at the funnel domain and the porter domain and we compare it to ACRB, it looks pretty much the same, right? At the ground state, it's like 71% similar across the protein, and they look pretty much the same. So they both have these asymmetric states. So in one protomer, they have access, binding, extrusion. So they still go through these quite extreme conformational changes, but they seem to do it in a similar way. But when we looked at the transmembrane region, this is when we start to see some interesting differences. So in the extrusion and the binding state, it looked pretty similar. But when we looked in the access state, which is when protons would first gain access to that transmembrane area to power that motion, is MdtF seems to already be swung out and trapped in a state which looks exactly like its binding state. So instead of it in ACRB, it goes from this access to binding to extrusion, conformational changes in the transmembrane region. In MdtF, it seems to be binding, extrusion state. So it seems to have a less kind of asymmetric transmembrane conformational change than in MdtF than it does in ACRB. And we can kind of look at this when we look at the flanking transmembrane 2 and 8, which are kind of the pistons which move the porter domain conformational changes. We see that in MdtF, there's just differences in the kind of up and down motions of these transmembrane regions in MdtF versus ACRB. So something seems to be different there. And we wanted to see if that was just an artifact, right, of, MdtF is just different. So we thought, how could we investigate this a bit further? What we thought would be quite interesting is there was a single point mutation which was found in MdtF, this V610F, where when they expose E. coli to an antibiotic levofloxacin, it kind of selectively pressurized it to make this mutant, which increased its kind of efflux profile, but made it more resistant to linezolid. So now we have an MdtF which is a slightly different kind of efflux specificity, but does it now also still seem to have the same kind of transmembrane differences to ACRB? And basically, there's a lot more detail in our archive on this, but essentially that seems to be the case. So this single point mutation seems to change the binding pockets in a way which makes sense for its change in efflux specificity, but it doesn't change the way that the transmembrane domains work. So MdtF still has this swung out state, whether or not the efflux specificity is different, if you see what I mean. But of course, this is a kind of ground state, right? So we try to make it wiggle and jiggle. We try to make it move. And so we collaborate with Attilio Vargu's lab to do molecular dynamics. And what we could see is when we focus on the porter domain is ACRB kind of, moves between axis binding extrusion states in this manner. When we look at the MdtF wild type, it kind of does it in a very different way. So it has very different contacts between these kind of different regions in the kind of drug acceptance and binding pocket region. And then when we have the mutation, we see that the mutation itself also kind of changes it and makes it more ACRB-like, which makes sense to the efflux specificity, right? So the mutant seems to cause something different where drugs kind of bind and get recognized in a way which makes it more ACRB-like than the wild type. But then when we focus on the transmembrane domain, what we see is, you know, cutting the story short, is MdtF mutant and wild type seem to be more similar than they do to ACRB. And the way that we measure this is we look at these really important residues for the protein relay network. So these collection of residues are essential across all these RND transporters to take protons, export them across, and power that motion. If you get rid of them, the protein dies. So we can kind of show this with this efflux assay where NAL-RED gets exported out. If you mutate this 408 residue to an aniline, it can't efflux anything anymore. It kind of dies, right? And so when we do molecular dynamics and we focus on the kind of hydrogen bonding contacts between these residues which shuttle protons, what we see is if we look specifically in this extrusion region here and the binding region here, is MdtF in blue and orange here seems to have a more engaged bundle in the extrusion and binding states than ACRB does. So that might be what's changing that transmembrane conformational change to power MdtF differently. And so we were interested in, does that reduce the proton exchange? You know, could that be the consequence for this conformational profile? So we did some kind of simple assays, these NAL-RED efflux assays, like I said, and we saw that MdtF does still export and the mutant exports in a similar way. But interestingly, ACRB seems to be much better than MdtF at exporting things at neutral pH, right? So ACRB seems to be a better pump than MdtF. And so we collaborated with the Helen Zagorska lab in Oklahoma to kind of look at this in more detail. And it just so happened that previously we were doing some other work and we looked at ACRB function across pH and we saw that it functions pretty similar from pH 5 to pH 8 and it functions pretty well. This assay is slightly different where we look at how much of a dye, which is an efflux substrate, remains inside the bacteria to how much gets exported out. So the kind of lower it is, the more export we're kind of getting outside the cell. And importantly, we chose to use a non-ionizable dye because when we look at pH changes, we don't want it to affect the drug molecule itself, right? We want it just to be a response from what's happening to the bacteria. And then when we did it for MdtF, what we saw is, you know, here's your kind of vector control across pHs. But what we saw is at kind of neutral pH, it was not that great as a pump, which kind of mirrored the other efflux assay we did before. But when we increased the pH, it got pretty good up to a similar level to what ACRB was. So when you start to go towards more acidic, so this is a kind of acid activated or acid responsive pump. ACRB is not that. ACRB is just a pretty good pump, right? It's like probably the best pump it has in doing this promiscuous level export. And what was really nice is we used these lovely pore cells, which the Zagorska lab developed, where we're able to kind of put a massive pore in the outer membrane to kind of eliminate this kind of permeability barrier. So we really are looking at efflux here rather than, oh, does it just kind of get in differently into this kind of bacterium or not? So this kind of led us to, oh, sorry.
Sorry, Catherine, I think we're getting towards the end of the time. So
I'm here. I'm done. I'm done.
Oh, great.
So this kind of gets us to the very end, which is what is it doing and why do we need it? So what we thought was interesting is when bacteria gets stressed and it remodels its membrane, it's kind of defending itself already with that remodeling. So having something like ACRAB there is kind of not that useful, right? Because it's not really dealing with attack in the same way. And actually, PMF dependent efflux pumps are pretty costly. So having a really good pump around is going to be quite costly. So maybe you could have MdtF, which, you know, is pretty more steady, right? You know, uses less PMF, pretty stressed, but it's still important for just disposing toxic metabolites from inside the cell, right? So not dealing with attack. But then when attack does come, when acid comes in, usually that membrane remodeling is not strong enough to deal with that. Then it turns on and it becomes just as good as ACRB. So you're kind of rigging the system, preparing yourself for acid attack. But MdtF is still doing a function to deal with toxic waste. But then when the acid hits the envelope, it really turns on and becomes really useful. So obviously in the future, we'd like to do this on other polymers which can deal with acid. And with that, thank you, because I've taken too long. Here's all the people. Thank you very much.
Thanks, Eamonn. That was great. I mean, I would love to get carried away and hear another 20 minutes of it. But okay, so Michael has a question. Michael, do you want to just jump in?
Just wondering if you would prefer a different polymer that didn't have any charge that would create confounding issues with ions and acidic phospholipids. Dibben would be great, but would there be any other properties that you want?
Yeah, mainly acid stability. I mean, I think I would be interested. We did some other data which the periplasm actually has high concentrations of magnesium because you have the Donen potential. So it actually concentrates magnesium in the periplasm. So exploring the effect of magnesium concentrations on the protein as well be interesting. Truthfully, we did this assay after we got the structure. So it's one of those things where the reviewer is like, why do you not do this at pH 5? And we were like, well, because that's not how the timeline worked, right? But it still does function in neutral pH, this protein, right? So it's still important to understand that state, but it would be really interesting to do a disc in acid to see what happens. Yes, definitely.
Okay.
Okay, great. So I think maybe in the interest of time, we'll move on to the next speaker. But if anyone has further questions, I certainly do. Might pop something in the chat. Thank you. That was great.
Thank you.
So our next speaker is Frank Bernhard. So Frank, yep, I can see you there. Are you ready to go with your slides?
Okay. So you hear and see anything?
Yep, we can see your slides.
Okay.
Looks perfect.
So I'm going to present you our process based on cell-free expression to produce gravity structures of membrane protein complexes in nanoparticles. I particularly focus on the process, on the protocol, and I show you two important tools. First, to get reliable yield and reliable quality of your samples. And I demonstrate the process with cryo structures of a number of GPCR G protein complexes. So we use a two-compartment cell-free system based on E. coli lysates. It's a coupled transcription translation system, so we just add plasmid DNA. Expression is based on the T7 promoter, and we use a complete in-house system. So we make our own lysates, our own T7 polymerase. Our lysate is partially purified, so we remove about more than 90% of all membrane proteins and about 50% of the soluble proteins. And we also make our own reaction containers, so small and prep scale. And so we have complete control over the whole process of cell-free expression. I think that is very important to receive reliable and reproducible results. Our strategy is to co-translationally insert the membrane proteins into preformed empty nanoparticles, in that case nanodiscs. We could show that during the insertion, a number of lipids are simply kicked out. Yeah, so the discs do not expand as they stay compact and they have a stable form. So we preform nanodiscs. So we fill them with different lipids or lipid combinations. We add them in stoichiometric concentrations to our cell-free reactions. So the ratio of the synthesized protein to the supplied nanodisc is usually 1:5, 1:6. And the advantage of this strategy is that the insertion is completely translocon independent. You're not limited by microsomes or any other translocon containing compounds, and you have complete control of the stoichiometry. So the first problem you have is to get sufficient yield of your protein. And here the good news is that this usually you obtain instantly because the protein production in the cell-free system the complexity is largely reduced to the basic translation process. We have the strong T7 promoter, so we don't have any problem with transcription. We can take care of the coding sequence by adjusting it to the E. coli codon usage. So the only remaining major problem is the initiation of translation. It's well known that the ribosomal binding site needs to interact with the ribosomal RNA. And this only happens if it's freely available and also if the start codon is freely available. And as soon as these are kind of involved in secondary hairpin formation with some sequences downstream, you have bad or even no translation. And this is basically not predictable, and we can easily address this by adding small so-called expression tags just immediately downstream of the ADG translation start codon. This is a small screen, so we just took a number of small expression tags. They are never larger than 6 codons, and they are common with having this AAA codon, which is very efficient in promoting translation instantly after the ADG start codon. And here you see the effect based on 5 GPCRs. You see that the white bar is the non-tagged GPCR, the expression of the non-tagged GPCR. And you see that some of them are quite well expressed without any expression tag. But these two, the GLP1R or the CCR3, are basically not expressed with a native sequence here, and they can be boosted tremendously by adding such a small expression tag. So we routinely add this tag when you have a new target. And usually we always get instantly a decent, sufficient expression. There was a recent report by the Kim group who confirmed these results. Also, they checked the number mostly of GPCRs, and they had many which did not express at all. And just by adding such a small expression tag, they came basically from 0 to high-level production. So this is one very efficient, important tool. The second challenge you have is to obtain a good protein quality. So when we co-translationally insert our membrane protein into the nanodiscs, it should fold, completely insert into the membrane and fold completely. That is what we wish. But unfortunately, what we get is usually also additionally membrane proteins which only partially insert into the nanodisc or just attached to the nanodiscs. They're also soluble. And so they cause a problem because how to keep apart these unfolded solubilized membrane proteins from folded solubilized membrane proteins. It not always helps to attach GFP to these proteins because we notice that GFP very nicely folds independently of the conformation of the attached protein. So we often also have even precipitates here of membrane proteins nicely with nice fluorescence. So FSEC, which you might know from classical cell-based expression, does not work as well with the cell-free system. However, we noticed that the unfolded or only attached membrane proteins to the nanodiscs can be nicely separated from the completely integrated nanodisc by single size exclusion. So we call this NSEC, nanoparticle-based size exclusion. So the idea is that the empty nanodiscs and the nanodisc with a completely integrated membrane protein do not change in the hydrodynamic diameter. So the size or the diameter is still just determined by the size of the nanodiscs. So even if you have an insertion of a very large protein, like here with proteorhodopsin, the size of the nanodisc does not change, and we always have illusion at roughly 1.5 milliliter volume. Here also you see the tumbling of the histamine receptor inserted into the nanodisc, and even with the attached G proteins, the diameter of these particles still is more or less eluting at 1.5 milliliter. So this is a very valuable tool to separate unfolded protein from probably folded protein. It's a prerequisite to get this 1.5 milliliter peak here for the functional folding of your protein. So you can use this easy and fast ENSEC tool to rapidly improve the quality of your protein. We checked that the 1.5 peak contains folded protein by several assays, so GPCRs where we had functional assays. So for the beta-adrenergic receptor, we had a ligand-binding radio assay, and you see that only activity, or the most activity, is present here in this peak 2, and you almost have no activity in this peak 1. And two other assays, we made a pull down of the histamine 2 receptor with this cognate GS protein, or a pull down of the self-resynthesized endothelin D receptor with immobilized agonist ET1. And in both cases, the elution profile of the functional protein is at 1.5 milliliter. This confirms that in this large first peak, you virtually have no functional protein, and all the functional protein is located in the second 1.5 milliliter peak. So the key point for sample optimization is to bring this first peak down and to bring the second peak up. And you have a lot of tools to optimize the quality and the stability of your synthesized membrane protein. Just considering all these different lipids, you can fill your nanodiscs with a lipid combination. So you can add agonists, ligands to the cofactors to the reaction. It's just exemplified here for the histamine 2 receptor. Here you see the quality of the histamine 2 receptor in different lipids, and you see that some lipids you get quite a decent functional peak, with others you almost have no functional peak. Here's the effect of added ligands into the cell-free reaction, and you see that in particular antagonists have a very strong positive effect here on the quality of your synthesized histamine 2 receptor. So that's the way how we optimize the samples. It's principle very easy and in a routine assay, and I'll show you the results now with cryo structures of 3 different complexes. That was the histamine 2 receptor, the beta-1 adrenergic receptor, and the F3 fatty acid 2 receptors. All of 3 of them are quite important for some human diseases. Here you see the ENSEC profile, the final ENSEC profile of the 3 receptors. That's what we used to obtain the cryo-EM structures. The free fatty acid 2 receptor is quite homogeneous. You have a very strong peak, 1.5 peaks of functional protein. However, beta-1 and the histamine 2 receptor, you see that the functional peak is in the minor fraction. So that was completely okay. That is completely sufficient to obtain cryo-EM structures. So our final samples had about 3 milligram per mil, and you only need about 30 microliter of that sample. Was completely sufficient to obtain a whole data set for the cryo-EM structure. Here you see ENSEC profiles of some other GPCRs, and you see that some of them, these 3, look quite okay, even better than the histamine 2 or beta-1 adrenergic receptor. Just if you compare these two, these are 2 subtypes of the same family, so more than 50% identical receptors, and you see that that one has almost no functional protein. It's solubilized, we have a nice solubilization of this protein, but if you make the ENSEC profile, you see you don't have any completely folded protein. In contrast, the other subtype here is very nicely has a large folded peak similar to the free fatty acid 2 receptor. So you see that even very closely related GPCRs can behave very different. Then when you have this protocol optimized, the rest is basically just routine. So you add your DNA templates with the reaction, the proper agonist, the proper nanodiscs, G proteins, and if you have a stabilizing nanobody. And then the procedure is you express overnight. Next day you make an affinity purification and the ENSEC purification. And then already in the afternoon, you can prepare the crits for cryo-EM. We have obtained the structure of the histamine 2 receptor with GS trimer. I don't want to go into detail of the structures, or they are published. Just one important point was that we could identify one aspartic residue in the ligand binding pocket, which is operating basically like a selective filter here for subtype specific ligands that only bind to the histamine 2 receptor subtype. The next structure is a beta-1 adrenergic receptor, same procedure. You just exchange the DNA template and the ligand in the reaction. And we obtained the structure with a similar resolution. There are many structures of the beta-1 adrenergic receptor available. However, almost all of them are from Turkey, and all available structures are heavily engineered. They have truncated termini and a truncated ICL3 loop. And the reason of that is because the human beta-1 adrenergic receptor was published to be very sensitive against detergent. And it is also unstable if it contains these dynamic loops. So it's a perfect target also for cell-free expression. So we could make the first structure of the full-length human beta-1 adrenergic receptor also in complex with a GS trimer. The structure is, so the interaction with the GS trimer is virtually identical in those areas which were published with a truncated version. However, we could find additional interacting regions. So if this large in the ICL3 loop is present, we have an elongated TM5. Yeah, so we could find here another interaction cluster with GS alpha. Furthermore, we could find a new interaction cluster with ICL2 to G alpha S, and we could find a new interaction cluster of the ICL1, even with the G beta 1 protein, as shown here. So overall, the interaction surface of the truncated compared with a full-length receptor is increased here for more than 300 square angstroms. So this shows here also the truncated receptor is functional. The full-length receptor has a different conformation interaction interface with the G proteins and a much stronger interaction with the G protein, resulting also in an increased signaling in the cells. The third example I want to show you is a free fatty acid 2 receptor. Here we were too slow, so we just came to 10 angstrom structure, and then the structure was published by another group. So we decided to not continue with the structure, but to rather focus on the dimerization of this protein. The FF2 receptor is reported to make homodimers as well as heterodimers with the FF3 receptor, and they do not dimerize with the FF1 receptor, which is recognizing a large fatty acid, and these are recognizing short-chain fatty acid. So procedure was the same, production procedure, DNA template, then acetate as ligand, same purification procedure. And what I want to show is just one dimerization assay, also implementing a new technique, the so-called nanotransfer, which combines the cell-free expression with cell-based assays. There are plenty of techniques here to transfer soluble proteins into living cells, yeah, but nothing to transfer membrane proteins into the membranes of living cells. And this we can address now by the nanotransfer technique, which is also a very easy and fast technique. You simply mix your living cells with your purified membrane protein nanoparticles, and the two membranes line, and within basically a few minutes here, the lipid and anything which is in this lipid is transferred here into the cell membrane. It's a very efficient and universal technique. You can also transfer, here we have a GFP fused GPCR transferred into primary cells, that's freshly isolated hard muscle cells, which basically cannot be transfected. Compared to the classical transfection, the difference is the transfer is not as efficient as transfection. In transfection, you have always a few cells expressing a protein like hell. That is not such the case in the transferred protein. However, the transfer is more uniform, so you have a much more uniform cell population because almost any cell is transferred at equal amounts. You can tune the transferred protein just by longer incubation or by incubation with higher concentration of your nanodisk particle. The transfer is completely independent of the lipid, of the membrane composition of your nanodisk. And it also is universal. Here we check the transfer of a number of different self-resynthesized GPCRs. What I want to show you is an interaction of our self-resynthesized FF2 receptor with a FF2 and
Frank,
FF2.
sorry to interrupt.
I'm done in a minute.
Okay, great, thank you.
With the FF2 or FF3 receptors synthesized with transfected cells. So the green GPCR is coming from inside, the blue one is strep-tagged and coming from outside. Here is just the control, so that is a transferred strep-tagged protein. That is a cell-synthesized flag-tagged green protein. And here we simply made a pull-down. We could show the SF2 receptor is the cell-based synthesized FF2 receptor is nicely pulled down by the transferred FF2 receptor, and here it's the FF3 receptor by the FF2. So here we have the homodimer, and here we have the heterodimer. This nicely shows, that the self-resynthesized GPCRs act the same, they have the same interaction like the cell-based synthesized GPCRs. So that's the end. I have some memos here. So for reliable expression, just simply take small N-terminal expression tags, NSEC control, very valuable tool, independent of membrane protein type or function. For quality monitoring, E. coli lysates are perfect suitable for complex eukaryotic membrane proteins. And you can use a nanotransfer to combine self-reproduction with cell-based assays. That's our cooperation partner, of course. Most of the work was done by other groups. That's our cooperation partners for the structural evaluation, and that's for the functional evaluation. That's our funding organization. And also I want to point out, if you are interested in our technique, we offer support in training, consulting, transfer of materials or protocol development. Just contact us. And I thank you for your attention.
Thank you very much. That was fascinating as well, especially with the transfer of proteins back into cells. Really, really exciting. I don't think there are any questions, and I think we're getting towards the end of the session. So could I invite the next speaker to bring their slides up? And maybe whilst that's happening, if there are any really pressing questions, we could take one or two. So finally, of course, Michael is our next speaker. Talking about the language of lipids and proteins and improved SMA derivatives.
Almost there. Thank you for the introduction. I'm going to talk about a recent paper that we've just published, but also some new ideas as well. Let me just share this, and then hopefully I'm good to go. I want to thank you also for chairing today. That was great. I've learned a lot today, as usual. And okay. Okay, maybe that looks okay there. And then
looks great to me here.
Okay, I'll just use this pointer if that's okay. Maybe we can go here. Okay. All right, so I want to talk about this language of lipids and proteins. And this is a depiction of Edmonton. One side of the river is really busy with buildings. Those are like the proteins. Most people study proteins. But we also live on this other side where the lipids are. And there are a few bridges that cross, and polymers provide a bridge between these two worlds, as does language. It allows us to have a common understanding. So I want to go back to the good old days. Here is Francis Crick reporting on the discovery of the central dogma with the linear transfer information from DNA to RNA to protein. So this is information transfer in sequences. And this was presented about 69 years ago. But this is not the full story. This doesn't encode life. It just encodes proteins. So if we want to understand how the entirety of a cell or a living organism is encoded, we need to go further. And I would argue that proteins encode the information necessary to produce membranes, and that this represents, at a simple level, the complete dogma that allows DNA to encode a cell, and when that's repeated n times, then that encodes an entire organism, such as yourself. And so we really need to understand better how proteins encode membranes. And there have been many models of membranes over the years. One of the first was the fluid mosaic model, where these integral and peripheral proteins are burying their hydrophobicity in a fluid lipid bilayer. This was followed by a mattress model with matching hydrophobicity of proteins and lipids that segregate to them. And then the lipid raft model, where lipids segregate into rafts and proteins preferentially are recruited to those rafts or excluded from them to create this heterogeneous plasma membrane organization. And then there was the membrane cytoskeletal fence model, where cytoskeletal proteins form rigid fences that constrain the diffusion of these transmembrane proteins and the peripheral proteins that are attached to them. And then a lipid fingerprint model from Tielemann's group, saying that lipids asymmetrically associate in a unique manner with each transmembrane protein. And then another paper recently showed that SMA could be used to excise a disc along with the associated lipids, and this was referred to as the prey protein lipidome. So lots of models. And then in my lab, we've studied how proteins associate with membranes. So this is a 5-domain from the EA1 protein. And we would argue that it first interacts with the membrane through its dipole. So dipole attraction of its basic surface with the acidic phospholipids found in cytosolic membranes. And then there is an insertion of a hydrophobic tow into the membrane via these residues with methyl groups or sometimes aromatics. This allows the protein to skate on the surface of the membrane until it encounters a lipid such as a phosphoinositide. And these are recognized stereospecifically. So here's the PI3P head group, a sugar head group with a 1,3-phosphate that are recognized by arginines, for example. In the zoo model, you can see the details as well as the zincs that stabilize the structure. And then we also found that other lipids also are bound. So here's the PI3P site, and then in green is where phosphatidylserine sits, and we could see that by NMR as well. So there is both dipole attraction, insertion, stereospecific recognition, and coligation. So you could call this the disc rule. But all these models are partial. So we're just looking at part of the elephant, part of the membrane, and we don't yet have a really holistic view of how the membrane gets organized in all its different compartments and complexities. So let's go back to the basics of the genetic code, which is now pretty well understood. And it really can be boiled down to the concept of a codon, where you have triplets that specify each of 20 amino acids as well as stop signals. So here's one triplet that is read by the ribosome to encode arginine, which is added to the growing polypeptide chain. So it's a very elegant, symmetric model that's simple, efficient, and accurate, as it needs to be to have been optimized over a few billion years and used by basically all organisms. And I'm arguing that membranes are similar in the sense that membranes typically offer a unique combination of a stereospecific lipid, a polar lipid, and a hydrophobic lipid. And these constitute what I would call the lipidon, or the lipid codon, which is a unique set of lipids, a barcode that is read by a protein. And that allows that protein to localize to part of the membrane. So that also encodes then the location of activation of this protein via the reader. And so rather than having 3 nucleotides encoded in amino acid, we have 3 lipids that are co-located, not covalently bound, and they specify location, not the amino acid addition. But it's the same kind of simplicity. And again, these need to be simple codes, and they need to be very energetically efficient. So we know that there are various lipids that are localized to different compartments. So we study this phosphoinositide with a 3-phosphate here on this position of the anositol ring, which is uniquely found on the early endosome and downstream compartments. And there are other lipids as well with different phosphorylation states, as well as the kinases and phosphatases that regulate their production that are also localized differentially to these different compartments. So localization is well known. And it's also now known that there are thousands of human proteins. We published this a few years ago. We identified about 1,900 proteins that contain 70 different domains that are known to recognize phosphoinositides. For example, the PX domain is found in 49 human proteins, and the different members recognize the 8 different phosphorylation states that are color-coded here, as in the previous slide. And here's a few 3D structures of different proteins, PX domains, plex domains, C2 domains, with different phosphoinositides bound, their head groups bound stereospecifically. So this then can be boiled down to what we would call the biological code that consists of both the genetic code and now the proteolipid code. And the genetic code is well known. At large scale, it's the conversion of nucleic acid information to protein information. And we would argue that the protein information that encodes membranes via these recognition of the lipidon features. There are also protein-protein interactions, of course, that are also key. And at a medium resolution, then you have genes that are encoding domains, typically, of protein structure. And then these are involved in forming zones, which are functional units of membranes. And at the lower level of resolution, you could say these are consisting of units of nucleotides, amino acids, and largely lipids. And so this is a simple code. The new features here are that we're talking about now proteolipid codes, which are instructions for membrane zone formation based on specific recognition of lipid combinations by protein molecules. And zones are then the structurally and functionally unique contiguous regions that define each type of membrane. And I would argue this should replace lipid rafts, which I think are frankly naive, because in a cell, lipids don't really self-associate. They're driven to their locations by proteins. I mean, they do self-associate, but they don't segregate as they would in a liposome. And then nanodomains and microdomains are ambiguous. We prefer that domain be used for protein structure rather than membrane regions. So we're hoping that zones becomes the default term. And zones have a structural hierarchy. There's the primary attributes, which is the combination of lipids in a particular ratio, and then a protein, which could be lipidated. You can also have trimers or tetramers, for example, higher order complexity. The secondary attributes are the arrangement in space, the contacts between domains and lipids that become more structured as they associate. They can also be transmembrane or peripheral. The tertiary attributes refer to curvatures, for example, constrictions, gradients, and waves, so more macroscopic features. And then the quaternary level is the interactions between zones, for example, during membrane fusion or fission reactions. So again, like a protein structure, there is a hierarchy of different structural attributes that allows this to be easier to understand and manipulate. And then I talked earlier about the phosphoinositides that decorate different endosomal compartments and also the plasma membrane. But there's also other lipids that uniquely decorate the ER and the Golgi and the mitochondria. So these form part of these zip codes or barcodes, if you will. They're present in the lipidons. And there are many other lipids. These are the 23 major types that impart some localization information. And then the enzymes and transferases that also are interconverting these, as shown by arrows, are also compartmentalized differentially. And so this then yields a cyclic biological code where you have replication, transcription, translation happening. This is happening on membranes. Membranes are also organizing the folding process. The ribosome is sitting on a membrane typically. And then these proteins that are first produced in an immature state then can be zoned to, for example, a downstream membrane organelle. And this then organizes the functional structure, which can then lead to the biogenesis of organelles in a cyclical manner. And then this is repeated for each cell. And the lipids can also, of course, be glycosylated, as can the proteins. This leads to further ramification of the code. And then looking at the cell again from maybe another perspective, you can see that the different organelles don't just have different lipids decorating their surfaces as indicated by colors, but there's also gradients of ions, for example, protons to the acidic lysosome, also of calcium ions on the plasma membrane, for example, by channels, and then also of lipid modification. And so it's both the lipids in these compartments, their modifications, and their ions that they bind to that triangulate where proteins go to in a cell, and then other protein-protein interactions are superimposed on that membrane organization. And the protein organization also, we know, is very complex in a membrane. There's various modes. I show 11 here. We typically think of a single transmembrane helix in a bitopic protein or a polytopic protein like this GPCR with 7 transmembrane helices. But these proteins are also, for example, palmitoylated. In this case, there's a palmitoyl here and here. And then also it has peripheral domains. This extracellular domain has a membrane binding surface that we could predict. Also a 8th helix that probably sits on the membrane via its amphipathic character. And there's various other modifications, also disordered lipid binding regions. And so if you look at all these different modes of membrane binding, firstly, most proteins bind membranes through multiple mechanisms. And secondly, most proteins are in fact engaging membranes by either crossing them or binding peripherally or inserting into one level or just being repulsed by membranes. You could argue, in fact, that all proteins engage membranes one way or another. So to look at this complexity, we decided to focus on one family, the PX domain family. There's 49 of them, and we group them by color here that indicates the specificity. So the golden ones are absolutely specific for just one phosphoinositide, and these other ones have lowering degrees of selectivity for lipids. And then we found that the modification sites in purple localized to the membrane binding surfaces in red. And so there seemed to be a co-location. And this is shown here where the PX domains that are the most specific for phosphoinositides in gold are the ones that have the highest pip stop. Now, pip stop is nothing other than a serine, threonine, or tyrosine that's phosphorylated and is near the membrane binding surface. And so basically, the PX domains that are the most specific are also the ones that are most frequently known to be modified by phosphorylation. And the same is true of arginine methylation and lysine acetylation. These are residues that typically bind to the phosphates on the phosphoinositide or the phospholipid, and these tend to be more modified in the most specific PX domains. And so the cell has been evolved in order to regulate these membrane binding surfaces, in particular lipidon binding surfaces, we would argue. So I'll go through a couple of quick examples here. This is the SNCS1 sortinexin protein that has a PX domain, also a bar domain, and it then associates on the ER with this primary zone structure containing the sortinexin molecule as a dimer. It may also be myristoylated and it's palmitoylated, so it's got multiple means of membrane engagement. And it binds non-specifically, we would argue, to a set of lipids. And this then forms the inactive conformation that can't curve membranes. And then when it is translocated to the endosome, it then specifically engages the lipids that are there, the PI3P, also a higher concentration of phosphatidylserine and cholesterol. And this then induces the active conformation that is like a banana shape that can curve the membrane. And the principles of membrane binding are shown here. So here you have the PX domain, which is the business end, and there's a dipole. This is the dipole interaction, a basic side shown in 2 different perspectives, the 2 sides of the protein domain, and then the acidic cap. And this allows orientation and directionality to the membrane complex. There's a specific recognition of the PI3 lipid head group. And then there's also a groove that binds the polylipid phosphatidylserine. And there's also a pip stop. This is this residue here, a serine that can be phosphorylated to turn the complex off. And then if you look at the adjacent elements, there's also a cholesterol engaging amphipathic helix here. Also additional phosphatidylserines are bound by these lysines, we would argue. And when this forms this complex with the bar domain, it can then bind and tubulate the endosomal membrane to form a trafficking structure. This is another protein from yeast. The previous one was a mammalian protein. So PIL1, in the same way, can form a banana shape. It binds a different lipidon with PIP2, as well as PS and cholesterol, and they're shown here color-coded. And there's also, again, a dipole interaction with the acidic membrane surface via this basic surface of the protein. And this can then form a larger banana shape with more multimerization that can curve the plasma membrane into furrows. This is another protein, a bacterial one, that binds a different set of lipids. There's phospholipids that are exposed in the extracellular leaflet, and this causes instability. And so the SLY-B protein recognizes this as well as the LPS. It's palmitoylated and has DAG molecules. And so this combination then allows this 11-mer to form to protect the outer membrane, also to protect the beta barrels like BAM-A that are sitting in the outer membrane that are important for its functional and structural integrity. And so in conclusion, then, the proteolipid code, we would argue, instructs zone formation via these specific interactions, and it's zones that are the structural and functional units of membranes. They're defined by a convenient hierarchy of structure. And these lipidons contain several locally concentrated lipids that provide stereospecific recognition, a polar interaction, and hydrophobicity that uniquely stabilizes and activates the protein on a particular membrane destination. And this recognition is regulated often by pip stops and met stops that are added to the binding surface for that lipidon. And so even if you can't see the lipid, you can see these modifications by mass spec, for example, and that's like a shadow, if you will, that allows you to identify lipidon binding surfaces. And then these zones can be purified and analyzed in schmalps, and we're developing new polymers. If I can have one more minute or so, I'll just show you. There's a whole number of maybe a few dozen polymers that have been developed and used to solubilize different protein structures in membrane sections. We've been developing a novel polymer based on the feedback from this community that is fluorescent, zero-ionic, and also has minimal hydrophobicity. And it's different in the sense that it has an amine oxide, which is also what you find on LDAO. So that removes the net charge, but maintains the polarity. It's intrinsically fluorescent, so you can see the polymer at about 420 nanometers. It's stable and also concentration dependent, so you can measure the concentration of the polymer in your sample by its fluorescence intensity. The charge-charge interactions that plague some SMAs have been removed because these have zero-ionic side chains, and thus you could use it with different, like calcium, for example, at any concentration. Calcium can be used up to hundreds of millimolar or other cations. It's also less sticky because these are alternating. This subunit is repeating, so there's a 1-to-1 ratio and less stickiness. It's also more regularized. And these are then soluble and active across the pH and ion concentrations that you might want to use. And then we've used this to purify A2A receptor and PAGP beta barrels, which can be seen by SDS Page and Western blots. We have a library of different polymers that we're developing. Typically, these are good at about 1% for solubilization of membranes. And you can see that there's a consistent size of about 28 nanometers for the resulting discs that you can also see by EM. Thank you for your time. I'd be happy to take any questions, and really appreciate everybody coming on board and